that soil formation process start quite unexpectedly soon compared to natural soil forming processes. And they follow certain trends, so we were trying to assess that as well. These are some of the partial aims that we were trying to do during the study, and most of them were two instances of comparing directly the soil under 3 species that were planted at the same time, so like a common garden experiment. They follow certain trends, so we were trying to assess that as well. These are some of the partial aims that we were trying to do during the study, and most of them were two instances of comparing directly the soil under 3 species that were planted at the same time, so like a common garden experiment. Certain trends, so we were trying to.
Somebody wanted to interrupt you.
I can try beatboxing to this study, and most of them were two instances of comparing directly the soil under 3 species that were planted at the same time, so like a common garden.
I believe we can hear right now the presentation from room number 1. But please be patient. Somebody wanted to interrupt you.
Ale nie ma, YouTube jest wyłączone. Co to kuźwa jest?
Nie, przeglądarki nie włączajmy w ogóle.
Dobra, wyłączone.
Okay to proceed, right?
Yes, you can continue, but you have to switch on again your presentation.
Sure. So we've covered this part and this part. These are some of the partial aims. So we were trying to assess the rate and type of certain soil forming processes in the mining sites where we were doing it, and try to compare the rate of soil development at various stages. And we also tried to assess the effect of slope on the process of soil formation, but that did not really have conclusions which are worth it. So my study area is located in the Sokolov mining district of the northwestern Czech Republic. It's one of the most extensive coal mining districts there. And I focused my research on these 4 locations: Lokecka spoil heap, Bohemia 1, Antonin, and Litov spoil heap. They were all established during different times, and they vary in vegetation and time of establishment, but they all come from the coal mining region of this area. The methods for the research vary from like paper to paper and analysis to analysis, so I did not include them in this presentation. But to begin with, my former supervisor had an idea that in order for me to get familiar with the topic, I need to do some review papers and gather some information on it. So these are just 2 publications that we managed to put together. And from those, me, who wasn't initially in this part of the topic, managed to get some general ideas of the problems that post-mining sites and coal post-mining sites in particular are facing. So there are some big physical, chemical, and biological issues that may or may not be familiar with. The biggest issue on post-mining sites is how to successfully establish vegetation, obviously, if you're trying to revegetate them. And through this, I gained some information on some common tree species that are used for the process of reclamation, restoration, remediation, revegetation. There's very big confusion about the terms there. It's also explained. And there's nowadays an ongoing big debate about passive versus active revegetation practices. Active revegetation practices are mostly considered to be technical reclamation, which is also included in legislation of many countries. And nowadays they're debating whether that's good or bad, because some research have proven that passive revegetation, which means revegetation by natural succession processes, is sometimes much cheaper and much better for mainly biodiversity, but some other things in the long run in general. And regarding soil formation on post-mining sites, there was a debate about the speed. It really depends on the parent material of your post-mining sites in general. But sometimes the general idea is that soil formation on post-mining sites can sometimes be much faster than in nature because the pedogenetic parent material is already turbated and started weathering in some way. So the soil is a little bit more prepared to start the process of soil forming there. There is also a debate when you look at the literature about the so-called biological versus pedological soil development. So biological soil development is mostly based on observing the organic matter and nutrient changes and cycles, whereas pedological soil development is generally found to be you dig a hole in the ground and try to see which horizons are there. So through the formation of diagnostic soil horizons. However, that usually takes ages to have them. So most of it is the biological soil development observations. And there's also been some current trends in post-mining research, like countries which are leading the research, etc., mainly focusing on carbon sequestration and nutrient flows. Now, for Sokolov region, we sort of had an idea and a preliminary hypothesis that our soil will tend to evolve from pure technogenic post-mining technosoil to maybe a forest cambisol under different forests and trees. So, some of the main parts of my research you can find in these two publications. Number 3 marked here focuses on just the soil aspect of soil formed under different trees of the same establishment age some 50 years ago in Sokolov region at Antonin Forest Arboretum spoil heap. Both of these researches were done there. Just the number 4 was done in cooperation with the guys from the Faculty of Forestry of Czech University of Life Sciences. So they assessed everything like production potentials, biodiversity, and soil properties, which was my part and contribution to this research. So there are 2 of them. This one focuses mostly on soil. This one focuses on other aspects as well. And we found certain things there. So this is the Antonin Forest Arboretum there, established in the early 70s after the mining. And it's a really beautiful place to see. My research there was split. We tried to... this arboretum had over 220 different species and cultivars of different trees established there. So we managed to find 23 monocultures, like homogeneous stands that I can test the soils under. The broadleafs are marked light green. The conifers are marked dark green in this place. So these were some of the species that we've tested. All the broadleaf species were native to the area. From coniferous species, we had native larch, Norway spruce, and Scots pine. Everything else were introduced species there. And we've done some basic physical and chemical soil analysis on the soils under these trees. This is just so I wouldn't show you graphs, etc., which you can read in the paper. This is just some of the preliminary thin section microscopy showing you the porosity after 50 years of soil formed under European beech and under Norway spruce. And as you can see, the compaction levels under Norway spruce are much greater. You probably know why, because you are all somehow related to the forestry segment. Right, okay, so some of the main findings of the research, when you compared all the soil properties that were tested there, were that, as expected, some species like alder, beech, linden, pear, blue spruce, which is a little bit weird, and ponderosa pine have shown some good physical and chemical soil properties that are generally considered to be good. On the other hand, birch, larch, Norway spruce, mountain, Weymouth, and Scots pine did not do so well. And this graph shows, in a way, a split and the depth of the formed organomineral A horizon after these 50 years in the section. And you can almost clearly see the distinction between the conifers and the broadleafs. Like the conifers are on the left, under 3 centimeters of A horizon depth, and the broadleafs are mostly on the right with more than 3 centimeters of depth. Some exceptions here were blue spruce and black pine, and some exceptions from the broadleafs were hornbeam and birch. So this is just the principal component analysis of how they cluster and how they look, and there's also like a clear grouping trend between the broadleafs and conifers here. However, we did have 2 papers mentioned there. So what I showed here is just from soil properties perspective, from production potential and the actual health of the trees growing on at Antonin Forest Arboretum there. We have something like a conundrum. For example, Pitea pungens did really well from soil point of view. On the other hand, Pinus sylvestris did not do as well. From production potential and health of the stands there, Pinus sylvestris is one of the absolutely best growing tree species of the Arboretum, together with larch and some others. There's a potential debate that it also sort of depleted the soil properties and embodied it into their actual body structure. So that's one of the things, but it also shows why you need the sort of comprehensive approach, and you cannot just observe the soil without doing other things. On the other hand, Pitea pungens has a very reduced stand. It's dying off from climate and pathogen threats there, and there's just a few trees left there. So it's a thing worth taking home. In other parts, we try to assess the effect of time on soil forming processes. This paper has already been published. This one is under second round of review. So within this region, we took 3 stands of 3 localities of different age, these post-mining sites. So the youngest one was a monoculture of alders planted after some 30 years ago. The other one was a 50 years clear maple stand, and the third one is a very old mining site and one of the oldest in this region. It's around 90 years old, and it's a combination of cherry and maple. So what we did is we dug some deep soil profiles there and trying to assess the horizon boundaries formation, and took some samples of soil from these pits to analyze them in like their chemical and physical properties. The parent material of the entire region, apart from one spoil heap, is consisted of cypress clays, which are tertiary and quaternary clays, which you can clearly see this sort of leaf formation in them. And all of the profiles tested basically exhibited similar horizon formation. Organomineral, cambic, and then the parent material. They're basically proving our initial theory that it would shift from a technosol to a cambisol there. So this is just a picture showing 2 soil profiles from the oldest mining sites and their profile development. We dug in 3 locations, 6 profiles in total. One was always done on plain terrain, one was done on a slope, because we assumed that on plain terrain, the accumulation of organic matter and everything else would lead to actually slightly thicker horizon formations. So in the end, we got this. So 30 years, 50 years, 90 years. And you can see that with time, all of these horizon differences are sort of what we expected. The one on plain terrain here was thicker in terms of all horizons here. This one was sort of inconclusive. and this one was inversed. However, Loketska on level terrain turned out to have a slightly different parent material in the chemical analysis tested, so that may have led to slightly slower development. Basically, with time, we had clearer horizon boundaries, more observable color differences, etc., and wider ranges of physical and chemical properties results there. What we got from this is what's pretty much known. The effect of soil organic matter accumulation and mineralization did improve soil quality. We also tried to test the quality of humic substances, and the oldest one did prove to be better than the others. The effect of slope, it sort of confirms our initial expectations, apart, as I said, from this part here. And other part of this research that my other colleague is doing her PhD thesis on is lithof spoil heap over there. And lithof spoil heap is very interesting in terms of most of the parent material is the cypress clays, which generally have pH of 8 and above when you test them. However, this particular spoil heap contains some shales which contain pyrite, and pyrite has pyrite oxidation, so there are sulfuric acid flows, color changes, and dying off of vegetation. The lowest measured pH value there was 1.8, so see how hard it is. These are just some pine trees that unfortunately did not survive, so establishing vegetation there is pretty hard. However, we did a research. Some of our colleagues from our department did monitoring in like 100 points there in the region, and they did measurement of certain soil chemical properties in 1998. So we repeated this experiment in 2018, and we got some values from those. Generally, it should be published soon, we hope. But generally, an increase of pH, organic carbon content, and slight improvement in quality of humic substances was noticed in 2018. So after 20 years, the conditions got better in most of the region, but some parts of the region got more severe acidification over time, leading to some potential aluminum toxicity, etc., etc. From a forestry standpoint, afforestation, mainly by deciduous trees, supported the improvement of these soil properties as was expected. But the issue is it has high pyrite content. The species there were chosen wisely when they initially did it. However, it's just there's so much pyrite oxidation and sulfuric acid flows that it is a bit problematic in the end. So conclusions out of combined approach to all of this is that the effect of time is clearly visible, and accumulation of soil organic matter has a very important role, obviously. From the standpoint of soil formation and soil quality, some long-lasting broadleaf species and black alder, which is a commonly used species for post-mining sites, are recommended. However, some suitable native coniferous species like Scots pine and larch did grow well there. Spruce was a little bit more problematic. And some suitable introduced species were black pine, Serbian spruce, and Douglas fir, and all three of these grow in the Antonine Forest Arboretum extraordinarily well. The effect of relief on soil formation was noticed and addressed, but a little bit of further research is recommended. And we still have some future publication plans and some results which have not yet been processed. Now, I originally do come from Serbia, and we had Serbian spruce here. Serbian spruce is an endemic relic in Serbia and is slowly dying off and moving to some very small parts of certain mountains. And when I first came to Czech Republic and saw that they did a plantation and saw the closed canopy of Pizea morica here, I was really puzzled and I really liked it. So it also grows well, and some other people including my roommate here at the conference, were doing some research about Serbian spruce on this particular spoil heap, and the results are astonishing. So moving Serbian spruce from its native area a little bit to the north might be a way to preserve the species, and it works well sometimes in these conditions. Now, there are some other potential factors that we don't have time to assess, but here, the last slide is just a photo from a textbook we used back in the Faculty of Forestry, where an old professor of mine took a photo in the Sokolov mining region in Czechoslovakia at the time, and you can see his height compared to the established spruce trees. As we said, it started in like 72, 74, so this is how old they were in 81, and this is just a photo I took in the Serbian spruce forests in 2019, and this is the height of a man there, and you cannot see the tops there. So thank you very much for your attention, and if you have any questions, feel free to ambush me and ask me.
Thank you very much. All the questions will be given to you at the end of the session when we'll have discussion. Now I would like to invite the second person who will give his presentation. It is Karol Tomczak. So please come. He will talk about the growing silver birch on post-agricultural land for higher wood quality. It is a case study in northwestern Poland.
Hello everyone, my name is Karol Tomczak and I represent Poznań University of Life Sciences and also Łukasiewicz Poznań Institute of Technology. And today I want to show you some results from my PhD thesis, which is about physico-mechanical properties of birch, beech and oak from former agricultural land. But today we will focus more on dimensions and visual quality than mechanical strength outcome of cutting trees. So the study context is simple. First of all, it's my PhD study. So the second thing is that back in the days, after the Second World War, the afforestation of post-agricultural land was very intensive. Then also after between 80s and 90s last century, there was the several national programs around the Central Europe were established to increase the forest cover, and now we can get a groups of this planting, and we can check if post-agricultural land or other abandoned land like post-mining sites provide us wood quality which is suitable for wood industry and give us a source of wood which is not permanent forest stand, but it's other source which can be used for getting high quality wood. So my main objective was obviously to analyze the influence of post-agricultural land on the growth of silver birch. So we choose 8 study plots in northern Poland where birch feels good, let's say, and 4 plots were established on forest land and 4 on post-agricultural land. The history of land use, because we didn't do any soil comparison and check, we approved the history of land use by state forest documentation and also by using old maps from 19th century and 18th century. In total, we measured 488 trees. And we focused, there was a localization of the plots. They were mature trees from 70 to 84 years old. And we did simple measurements like diameter, height, crown base, crown length. And additionally, we measured the height of the first living branch and the height of the first dead branch to estimate the length of knotless trunk, which is clear from any roots because of the dead branch. So, and then we also evaluated the wood quality based on the visual defects. So we did an inspection of nodes, trunk shape, curves, and set the 3 grades: high quality wood, good quality wood, and pulp wood. And it was done based on the rules provided by state forests and which are used daily by local foresters in Poland. So let's come to the results. So as you can see, diameter on forest agricultural land was higher than in case of the forest land, but there was no statistically significant differences. In case of the height, we observed the same trend, that birch trees from former agricultural land was higher than trees from forest land, and then the statistically significant differences were found. When it comes to other trunk and crown characteristics, we can see that in most cases the former agricultural land had the greatest results, which means that obviously when we have crown base of tree which is set at higher, then the crown length is smaller. So however, when it comes to the stability of trees, when we check the slenderness ratio, it was more or less the same, so it didn't influence for the stability of trees. So let's go to the quality parameters. As you can see, the knotless length of the trunk, so this most precious wood, was outstanding on former agricultural land, which provide us a good perspective for high quality wood. And as you can see, visual quality provide us the same results, that higher share of wood, of high wood quality, was noticed on former agricultural land. And as you can see, the lower one, the pulp wood, let's say, also was the share was lower on the forest agricultural land. Okay, to conclude this, we can say, based on this case study, that there is a potential for wood production of broadleaf species, for example, birch, on post-agricultural land, especially in regions where the birch has suitable growth conditions. And this, I can spoiler you, that this perspective was confirmed in our next study, which consisted the mechanical strength of this tree which we cut. So even in that test, birch from former agricultural land was better. So as you saw, the birch from former agricultural land had a bigger dimension, and so the stand volume was also bigger. So it's a good perspective for wood production. And yeah. So if you want to get more information, you can read more in those 2 articles which were published last year. And from me, that's all. Thank you.
Thank you very much for the interesting presentation. And as before, we will wait with the question till the end of the session. And now I would like to invite Cornelia Amon to give her presentation. So now we will be able to listen and to see the results which are related to the statement protected forest patches conserve both carbon stocks and tree stand diversity. So Cornelia, the floor is yours.
Okay. Hi, my name is Cornelia and I'm from Austria. I'm from the Austrian. Thank you. Sorry. So I am from the Austrian Research Center for Forests, and I'm going to give you a talk today about how to protect both carbon stock and tree alpha diversity in protected forests of Austria. So at the Austrian Research Center for Forests, we currently have the Stepping Stone program, where we establish stepping stones all over Austria to enhance the overall connectivity of the Austrian forests. And we, in autumn last year, in 2023, we already established 188 stepping stones with 247 survey plots, which were ranging from 0.5 to 25 hectares in size. And all these stepping stones were registered voluntarily by forest owners themselves. So in the smaller stepping stones, we established survey plots that are like one survey plot that is representative for the stepping stone, and also with a buffer of 30 meters to the edge to reduce edge effects. And in the bigger stepping stones, we established the survey plot systematically with a 100-meter raster. We use this 30-meter buffer to the edge to reduce the edge effects to the survey plots. So as the forests, the forest ecosystems are very complex, there's a need to set priorities when it comes to the selection of appropriate stepping stones. So all the stepping stones were selected according to conservation targets, and these conservation targets were selected with structural characteristics of forests that are known to enhance the overall biodiversity in forests. And these are like forests with habitat trees, minimum 5 trees per hectare, special sites that are characterized by extreme conditions or the occurrences of rare species, and areas with a higher amount of deadwood, like minimum 20 cubic meter per hectare. All the stepping stones were also then assigned to forest types according to the Austrian National Forest Inventory and aggregated to the main 5. And these were coniferous forests that are mainly spruce fir and spruce and pine forests, acidophilous broadleaf forests that are pine and oak forests, hydrogenic forests that are riparian and black elder forests, broadleaf forests, which are mainly oak and beech forests, and mixed forests that are mainly beech, fir, and spruce forests. So on every survey plot, we measured the stand characteristics, and these were the tree species that are bigger than 5 centimeters at breast height, the tree height, all the tree microhabitats, and the lying and standing deadwood. And also in a smaller subplot, all tree species smaller than 5 centimeters, and also the shrubs were recorded. And all this happened, I forgot to mention, in a circular plot of 300 square meters in size. So my main research questions were, are there trade-offs or synergies between the above-ground carbon storage and tree alpha diversity in those stepping stones? And are there differences of stand structural characteristics among the aforementioned forest types and conservation targets? So the structural parameters were selected, inspired by Sabatini et al., and these were the biomass-related variables. It is above-ground biomass calculated by species allometric specific models, stand basal area, and standing deadwood basal area, and also lying deadwood volume, and as well as the leaf area index and veteran trees that are defined as tree species that are bigger than 50 centimeters in size. And for tree species diversity, the total number of tree species was recorded, and the Shannon-Wiener index, the name is then species diversity, to account for the abundances of the species. So for the variance analysis between the conservation targets and the forest types, we calculated the two-way ANOVA with the correction of Tukey posteriori test. And when we have a look at the results of the differences among the conservation targets, we had significant differences in above ground biomass, basal area, and lying deadwood. I only display above ground biomass here as basal area and above ground biomass is highly correlated with each other. You can see that stands oriented for the conservation target habitat trees had the highest above ground biomass here in blue, and stepping stones oriented for habitat trees and deadwood had twice higher basal area compared to the conservation target rare ecosystems. When we're looking at the results for forest types, we had a significant effect of the forest type on all stand characteristics, and we had the highest above ground biomass in hydrogenic here, in mixed and in broadleaf forests, and the lowest in coniferous and in acidophilous broadleaf forests. And when we have a look at the number of veteran trees, the highest number were recorded in hydrogenic and mixed forests. The leaf area index was the highest in hydrogenic forests, and interestingly, there were, and mixed was the second place, and there were no differences between tree species richness and diversity in a quantitative way among forest types. When we're looking at the principal components analysis and the relation of those structural characteristics to the forest types, to all the stepping stones, we can see that we have those two main directions. Like here we have tree species richness, and here all the biomass-related variables. That indicates that the preservation of carbon stock and tree species diversity should be treated as separate objectives when it comes to the selection of appropriate stepping stones. So before I'm drawing conclusions, I want to point out the limitations of the study. As these forests were registered voluntarily by the forest owners, they cannot be generalized over all forests of Austria. And also because they were selected according to conservation targets, but still they represent appropriate forests for conservation. And also, as we had to define this threshold of veteran trees that are bigger than 50 centimeters in size, we might have overlooked small veteran trees because they are not always correlated with size. The age is not always correlated with size. And we were unable to account for management-related historical data. So I'm coming to the conclusions. It is important to treat the preservation of carbon stock and tree species diversity in protected forests separately. Using the conservation targets simplifies the selection of appropriate stepping stones to when one looks for stepping stones with higher above ground biomass, and also when rare forest ecosystems have less above ground biomass, they have a high conservation value, and it's very important to include them. And also forest types have clear differences in above ground biomass, as we had the highest biomass in hydrogenic, mixed and broadleaf forests. And also when there's no difference of species richness in a quantitative way, still they have different species compositions, and it's important to have the wide range of different forest types to conserve all tree species. So thank you for your attention.
Thank you, Cornelia, very much for your presentation. Now we will move to the next point of our session, session number 7. Please, Katarzyna Rawlik. She will give her presentation titled How Do Herbaceous Forest Understory Plant Species Reflect Global Patterns? It is a study of the relationships between decomposition and plant functional traits. So, Kasia, please, the floor is yours.
Good morning. Thank you for introduction. In today's presentation, I'd like to show you preliminary results of my studies in project How do herbaceous forest understory plant species reflect global patterns, a study of relationships between decomposition and plant functional traits. The studies were conducted in collaboration with Professor Jagodziński in laboratory of Department of Ecology of Institute of Dendrology in Kórnik. So my studies are about decomposition and about functional traits. Decomposition, it's a broad term which includes physical, chemical, and biological mechanisms that transform organic matter into increasingly stable forms. Decomposition is important at the global level because it is responsible for huge amounts of the carbon dioxide returned to atmosphere. It's responsible for formation of humus, and it's closely tied to nutrient cycling. And functional traits are morphological, physiological, and phenological traits which impact growth, reproduction, and survival of organisms or population of plants. There is a general concept that plant life strategies impact the decomposition rate. According to this concept, plant species with conservative life strategies, resource conserving, decompose slower than fast-growing species with efficient resource acquisition strategies. These life strategies are connected with plant traits. For example, plant traits connected with conservative life strategies are high leaf drive matter content, low nutrient concentrations in litter on in leaves, and low specific leaf area. And these plants has a slower decomposition rate. However, many studies have demonstrated that plant traits have afterlife effects via their impact on the decomposition rate. It is not clear whether patterns found on a global scale are reproducible at local scales in specific growth form or in specific organs. A better understanding of this relationship between traits and decomposition is crucial for predicting plant responses to environmental changes, as well as the impact of individual species on ecosystems. On this picture, you can see the results of our previous studies on the composition of oak hornbeam species, understory species. And in these studies show that size-related traits like total biomass or total leaf area had a stronger influence on decomposition than economic traits, for example, specific leaf area, leaf dry matter content of litter nitrogen content. And these economic traits are commonly used as the best explanators of decomposition rates. Thus, the main aim of our study was to determine the rate of decomposition of 11 herbaceous species growing in different types of temperate forest, and to assess the correlation between the composition rate of necromass of these species and their functional traits. Yes. We hypothesized that the composition rate in our pool of species is strongly influenced by the life strategies of plants. and is positively correlated with specific leaf area as well as with biomass allocation to foliage. We established 9 research plots in central Wielkopolska, near Poznań. Distances between these plots these plots were established in 3 forest complexes near Poznań, and these forest complexes distances between these forest complexes are not less than 40 kilometers. We establish plots in 3 types of forest: pine forest, oak hornbeam, and ash alder riparian forest. These forests are quite well preserved and rich in tree species. The herb layer in these forests has a high degree of naturalness. And according to phytosociological approach, this type of forest, it was Leucobrio pinnetum, Gallio sylvatici carpinetum, and Fraxino alnetum. For our studies, we choose 11 vascular plant species, which are the most abundant herbaceous species in studied types of forest, and they grow along a wide gradient of soil, hydrologic, and light conditions, and are representing various life strategies: spring ephemerals, green color, summer green species, blue color, and grasses, red color. To know the composition rate of chosen species, we established the litter bag experiment. This litter bag method is commonly used in the composition studies. And we put known mass to labeled litter bags. We place these bags in forest on the forest ground. After no time, successively, some part of litter bags is collected, dried, and weighed, and analyzed for loss of mass. And in our experiment, for all species, we put into litter bags a mix of leaves and shoots in proportions similar to what occurs in specimens in the field. It was home plot experiment. It means that for each species, litter bags were placed in that type of forest where this species really exist. And moreover, we started the experiment at different dates for different group of species according to time of senescence of most plants of the particular taxa. Because we wanted to assess the composition rates in conditions similar to natural, as similar as possible. We assessed decade constants for each species according to single exponential model proposed by Olson. We assessed differences in the species studies decomposition rate by using ANOVA with post-hoc test. And to assess the correlations between plant traits and decomposition rates, we used non-metric multidimensional scaling. To characterize herb species traits, 10 individuals of each species were collected from same sample plots established for the composition experiment. We collected specimens at the time of flowering, and we assessed 12 traits, like total biomass, total above ground and below ground biomass, total leaf area, specific leaf area, leaf dry matter content, plant leaf and plant stem dry mass, below ground mass fraction, stem and leaf mass fraction, and height of the plants. Now just a look at the preliminary results. We found statistically significant differences in the rate of decomposition among species studied. Necromass of spring ephemerals decompose significantly faster than other plants, and especially two species of spring ephemerals, Corydalis cava and Ficaria verna. The necromass of grass decompose significantly slower. These species are Deschampsia flexuosa and Calamagrostis arundinacea. The decay rate calculated for studied species ranged from 0.3 for Calamagrostis arundinacea to 19.7 for Ficaria verna. The values of the decomposition rate obtained for grasses were comparable to leaf of trees of such species like Quercus robur, for example. And on different stages of the decomposition process, we found statistically significant differences between mass remaining between spring ephemerals, especially these two species, Corydalis cava and Ficaria verna. These species decomposed fast, and between grasses which decomposed slower. And we built up an ordination based on plant traits. And this diagram shows the differentiation of plant individuals belongs to 11 species. The main factor that differentiated these plant species are size traits, as stem and the leaf biomass. On the right side of the graph laid large plants, on the left small, including spring ephemerals. The grass species are quite different, and they are characterized by high stem mass fraction. And in the next step, we checked how the plant trait differentiation correlate with the decomposition rate. The composition rate correlates with the main differentiation gradient across first axis, and it's negatively correlated with plant size. Spring ephemerals, small plants, decompose the fast, and on the decay rate about 10. And on the opposite side of the diagram are grasses with K-decay less than 1. But these relationships, this relation between size and decomposition is not linear, as some small plants, for example, Oxalis acetosella, decompose slower than similar plants. And on the other side, Urtica dioica, which is big plant, decompose faster than similar plants. So just look at the conclusion. The composition is strongly influenced by the life strategy in a studied group of plants, and the composition rate is correlated with size traits, especially negatively with stem and leaf biomass in studied species. The study was financed by the National Science Center of Poland and partially by Institute of Dendrology. I would like to give a special thanks to Mateusz Rawlik, who helped me with statistical analysis and preparation of figure. And thank you all for attention.
Thank you very much for your presentation. Now we ask for the help our technical staff to switch on the movie. The movie contains a recorded presentation of Małgorzata Stanek. And Małgorzata Stanek will tell us about the comparison of leaf chemical characteristics between invasive monospecific, native monospecific, and native multispecific stands.
Hello, ladies and gentlemen. First of all, I would like to take the opportunity to thank the organizers for inviting me to the conference. Unfortunately, for reasons beyond my control, I cannot be with you in person, but I will be closely following the entire conference via the internet. I'm Małgorzata, and I work at the Institute of Botany of Polish Academy of Sciences in Kraków. I am also the head of the Ecochemistry and Environmental Engineering Lab in Sharp. Today, I would like to present the first preliminary results of the Invasive project. It has been running for a year and will continue for the next 2 years. First, I will give you some general information and then I will describe the Invasive project and the first results in more detail. Invasive alien plants may alter ecosystem functions and structure because they differ from native plant species in traits related to size, fitness, flow rate, physiology, leaf area allocation, and shoot allocation. They influence native plant communities directly and indirectly through their impact on soil environment. Invasive plants can alter soil properties through litter deposition and root exudates, which can modify soil nutrient pools and soil structure, as well as the activity, biomass, and composition of microbial communities and microbial groups, for example, mycorrhizal, saprotrophic, and pathogenic fungi. This in turn may change crucial ecosystem processes, such as organic matter decomposition and mineralization, element cycling, or nitrogen fixation. Alterations in the soil environment induced by plant invasions may lead to positive feedbacks that potentially stabilize or accelerate further invasion. Here is a simple representation of various factors that shape the microbiome. There are plant factors such as species and exudates, environmental factors, namely climate and soil properties, and finally, interactions between microbes such as metabolites and microbial hubs. Thus, all of the factors, the host, microbial interactions, and the environment are interconnected and jointly build the microbiome community. Biological invasions by alien species, those introduced by humans to regions outside their native distribution range, affect the majority of habitats, including semi-natural and natural ones, and impose high costs on the economy. According to Raymanek, 76 invasive tree and 61 invasive shrub species are reported for Europe, but Brass and others noted in 2019 that 153 species grow in European forests, which is about 4% of the total European forested areas. The largest areas are covered by Robinia pseudoacacia that occupies 2,44 million hectares in 29 countries. As you can see on the slide, some alien woody species are favored over native woody species due to their various positive features, for example, hardiness, faster growth rate, resistant to climate change. Compared to trees, little attention is paid to alien shrubs. Nevertheless, both alien trees and shrubs can have serious impact on species diversity and functioning of invaded ecosystems. On a large scale, they can be difficult to eradicate. However, recent studies reported the negative effects of frequent summer droughts and heat waves on economically and ecologically important native tree species in Europe, like Fagus sylvatica and Quercus robur. These species will likely lose their climatic niches in many areas that in turn may be invaded by alien woody species. Thus, comprehensive knowledge about the relationships between native and alien dominant woody species is needed to provide a sound basis for decision making on prioritizing the commercial use of alien tree species over environmental issues and vice versa. The project addressed the following question. What are the net impacts of the dominant species on plant communities, litter quality, microbial community, and nutrient availability? Is the distribution of some invasive and native species limited by soil conditions? What is the ranking of the studied species based on the overall impact they pose? However, today I would like to focus on the quality of senescent leaves collected at invasive monospecific, native monospecific, and native multispecific stands. So, we hypothesize that species containing more phenolics and tannins in their leaves would be less colonized by microbes and we expect that there would be more such species in the invasive group than in the native group. We also hypothesized that multi-species leaf material control would have a more constant chemical composition than single-species leaf material, both invasive and native species. For the study, 7 dominant invasive woody species and 7 dominant native woody species were selected. Based on the field observations, we choose species that form homogeneous patches. In the case of alien invasive species, these are Acernegundo, Allanthus altissima, Fraxinus pennsylvanica, Prunus terracifera, Prunus serotina, Quercus rubra, Robinia pseudoacacia, Spiratonitosa. While in the case of native species, these are Acer platanoides, Cornus sanguinea, Crataegus monogyna, Fraxinus excelsior, Populus tremula, Prunus spinosa, Quercus robur, and Tilia cordata. As we can see on the maps, the stands are located in the Czech Republic, south and west parts of Poland, and Slovakia. In total, 173 stands were established. Within them, 97 stands with invasive dominant and native dominant species, and 76 control stands with multi-species native vegetation. Here, you can see the scheme of the sampling at one stand and how the study plots were established. I need to highlight that monospecific stands of alien and native species dominants were located as close as possible to the corresponding multispecific stand control. In autumn 2023, senescent leaves from the dominant trees were collected separately from each stand. In the case of control plots, a representative mixture of leaves of all woody species occurring at each stand was prepared. At the laboratory, the content of carbon, nitrogen, calcium, magnesium, potassium, total phenolics, and condensed tannins, as well as microbial properties, were measured. So, let's move to the results. As you can see on the graphs, the leaves of dominant monospecific stands had lower content of total organic carbon and higher content of calcium and magnesium than the leaves on control multispecific stands. What is more, the leaves of invasive species had a higher content of total magnesium than the leaves of the native dominant species. On these graphs, we can see that the leaves of dominant monospecific stands, invasive and native, had significantly lower content of total phenolics and condensed tannins than the leaves on control multispecific stands. Moreover, the leaves of native species, of invasive species, had a lower content of total phenolics and condensed tannins than the leaves of native species. On the last graphs, we can see that the leaf chemistry differed significantly between species. For example, the Spiratomentosa leaves had significantly more condensed tannins, while Ailanthus altissima had significantly less condensed tannins than the other species. So let's sum up the conclusions. The leaves of dominant monospecific stands had lower content of total organic carbon and total phenolics, as well as higher content of calcium and magnesium than the leaves on control multispecific stands. The leaves of invasive species had a lower content of total phenolics and condensed tannins, and higher content of total magnesium than the leaves of native species. The leaf chemistry differed significantly between species. The correlation between phenolics and PLFA was not significant, as one of the aims was to explore the novel weapon hypothesis. This result could mean that this hypothesis may not be applicable to the species under study. The variance in the leaf chemistry for the control group was slightly smaller than for the invasive and native groups. Ladies and gentlemen, thank you for your attention and goodbye.
Unfortunately, she cannot hear the applause. Ah, yeah, okay. Because I think we have 2 minutes before the next presentation. If you have any questions, if Kasia is available online, so we can ask her. Okay, if there is no question, we can go to the stream from the room number 1, because as I told you before, Clayton Macado is not present today and he didn't deliver his presentation like Małgorzata did. So now we will move to the room number 1, and it's the exact moment when Tara Kaila should start her presentation, but we don't know if they keep the schedule. We should hear the presentation, which is titled Variation of Ion Concentrations in Stemflow and Truthful in Beech Forest in Germany. So please switch on the stream and we will see what they are doing.
Bars suggested high variability of value during measurements, and average for all days from leaf dry matter content is 8.8%, and specific leaf area is decreased 2%. Next chart show means changes and standard errors in relative water content expressed as the difference between the control and the test sample. The great difference in the decrease in water content are observed after the first day of application of increased temperature. The high of the error bars suggested low data variability. In the following days, we can say that there has been acclimatization and or stabilization, and average for all days is decreased for 3%. This chart shows means changes and standard errors in fluorescence chlorophyll and performance index under the influence of increased temperature. The biggest difference can be observed in the case of performance index for all days, and average for all days is increased performance index by 47%, and fluorescence chlorophyll by 3%. Despite such a large increase in photosynthetic efficiency, the total chlorophyll content did not change, as can be seen in this chart. Now I will move on the results obtained with ATR. This example chart, ATR show biochemical profiles of leaves yellow archangel. Blue is a control chart and red is a high temperature. I marked 4 peaks on the graph that showed a noticeable difference after the first day of the experiment. The marked peaks corresponded sequentially carbohydrates, next also carbohydrate, next pectin, and next also pectin. Based on the analysis loading plots, peaks were identified that determine the difference between the control sample and the test sample. For PC1 axis, explain peaks for carbohydrates, and PC2 axis for pectins. I marked these peaks on the previous slide. The first 2 PCA axes explain 47% and 23% of variance in the spectral data set. Non-metric multidimensional scaling based on the maximum peak absorbs values for a designated K-ATR range from the previous slide. C is a control group and T is a test group, and the number are the days of the experiment. The analysis showed that the sample were divided into 2 groups, control and experimental group. As you can see, the control group show greater diversity of feathers, which is visible in the form of a large circle. And conclusion, the yellow archangel is a good plant organism for studying the effect of temperature changes. There was a quick reaction to the change in parameters on day 0, specific leaf area, relative water content, performance index, and leaf biochemical compounds. On the following days of the study, the ecophysiological parameters constantly change. The increased temperatures is responsible for the increase in the dry matter content in the leaf and the decrease in specific leaf area. Increased temperature causes an immediate increase in performance index already on day 0. ATR results confirm change in the metabolism of plants adapting to higher temperature contact with carbohydrates. Each of the analyzed parameters reacts in a different way to temperature change. It can be said that the plant's reaction is complex. And thank you for your attention.
Many thanks to Beata for your interest in a very important study. And the last topic of our third session is about variations of iron concentration in stem flow in the beech forest, Germany. And I'm pleased to invite to Tara. You will make an oral presentation, please.
Hello, good morning everyone. My name is Tara Kaila. I come from University of Göttingen, Germany, and today I'm here to present my master thesis work for you. The topic is variation of iron concentrations in stem flow and through fall in a beech forest in Germany. This is my agenda for the presentation today. I want to briefly introduce about the topic that I'm going to present and explain about my research hypothesis and methodology and discuss the results and the discussion part and summarize the topic. Coming to the introduction, I would like to start with a short explanation of what is stem flow and through fall. I am sure most of you here know about it, but yeah, I just wanted to say what stem flow is. It's the intercepted rainwater that passes along the tree trunk towards the ground, whereas through fall is the rainwater that falls through the branches with or without getting in contact with the tree canopy. And why is it important to study about stem flow and through fall in forest? Because they play a huge role in nutrient cycling and water cycling in the forest ecosystems. And this is very important for the growth of the forest and also to sustain the forest in the long term. And due to this important role, we are interested in studying them, and we started our work with an objective to find the variations in the behavior of different ions in different partition fractions of rainfall. And here you can see the hydrological cycle of wooded ecosystems where stem flow, through fall, and other precipitation fractions are shown here. Before going into presenting my actual work, I wanted to tell about our work, which is really a long experiment. It started in 2017, and the first half of 2017 year was mostly for planning the work and just brainstorming about the objectives and what to do with it. And the next half was mostly for setting up of the infrastructure and starting of the experiment. In 2018, research and development work was done, mostly for collecting the data, rectifying errors, which was within the instruments that were set up in the field. And from 2019 to 21, very extensive sample collection and analysis was done. And in 2022, I started my master's thesis work for analyzing the data from the previous years and also in 2022. And I continued the data analysis with a focus on ion concentrations of stem flow, through fall, and rainfall samples. And in 2023, I submitted my master thesis. Our objective for this work, for my master thesis, for this work was to see how different ions in different concentrations was varying in these three partitions of rainfall. So to achieve this objective, we hypothesized that the larger the tree size, the higher the ion concentrations in the stem flow, and the ion concentrations of stem flow will be higher compared to through fall and rainfall concentrations. And there will be variations in stem flow, through fall ion concentrations between different climatic seasons. The main factors that we considered for my work are volumes and ion concentrations. The ions such as potassium, calcium, magnesium, sulfur, sodium, phosphorus, aluminium, manganese, iron, and zinc of stem flow, through fall, and rainfall. And we also considered meteorological seasons or climatic seasons for the third hypothesis. This is the experimental site. The experimental site is located towards the northern part of Germany in a place called Ebergotzen. And in the experimental sites, there were many trees, out of which I chose 12 trees for stem flow and 4 trees for through fall analysis. Within the 12 stem flow trees, I have divided them into big trees, small trees, and medium trees, and based on their diameter at breast height. The big trees are the trees which have more than 40 centimeters in diameter, and medium trees are 20 to 40 centimeters, and small trees are less than 20 centimeters. My experimental approach here was the setup was done, everything was done in the previous years, and the volumes data was collected using data loggers, which is automatically collected every 15 minutes intervals. And this 15-minute interval data is automatically in the data logger, which can be integrated into the SQL database once a day, and this is extracted through a R script which was done especially for this experiment, which is called Ebergodson. And the stemfall measuring resolutions with these tipping buckets here was 0.5 liters for big trees and medium trees, and 2.25 liters for small trees. And this collecting vessels is used for through-fall water collection. Iron concentrations was done on bi-weekly basis. For laboratory analysis, which was done by our department lab technicians, we used ICP-OES, inductively coupled plasma atomic emission spectroscopy. Statistical analysis was done by me. I used MS Excel for doing the data organization and the previous data setup, and our program version 4.2.1 was used to do the analysis using the following packages. For the data analysis, I did mostly ANOVA and regression analysis. Coming to the results, here are the results which showed that the first hypothesis showing the differences between iron concentrations between big, medium, and small trees. It wasn't clear pattern that it wasn't shown that big trees were higher in concentrations and small trees did not show any differences. We hypothesized in the other way, but this can be attributed that because of our methodology that we used, where we collected the stem flow volumes in a similar way for all the types of trees, we couldn't find any differences in the ion concentrations in any of the ions. Here comes the differences in stem flow, through fall, and rainfall. Our results showed that stem flow concentrations are significantly higher than through fall and rainfall concentrations for almost all the ions. Our results were in accordance with Johnson and Lehman studies which were done in beech forests, and this is already expected. And we attributed this to the canopy exchange and leaching of ions from the stem flow into the rainwater, and also due to high residence time in case of stem flow compared to through fall. And here is the volumes in stem flow, through fall, and rainfall in each year. It wasn't as expected, and it is not comparable to other studies, but there are also studies which had similar reports. In our case, we had way lesser stem flow compared to other studies, which was averaging around 7%. But there are several other studies in beech forest which showed up to 20%. This was due to the fact that we did on bi-weekly intervals instead of doing on a rain event basis. Here is the stem flow, through fall, ion concentrations in different climatic seasons. The concentrations were found to be higher in summer and autumn compared to winter and spring seasons. This was attributed due to the leaf senescence phase during these seasons. During this time, the mobility of nutrients is extremely high compared to other seasons. Thus, this can lead to a mobility of nutrients into the stem flow, causing high leaching. And in summer season, there will be high exposure of the leaf area, and also the contact surface for the leaching process will be higher, which can lead to more stem flow concentrations. I want to summarize that the effect of tree size was not evident for most of the ion concentrations, which can be due to the methodology that we have chosen. And the volumes of stem flow and true fall is not comparable to other studies, even though there are some studies which proved our research to be true, but this could be rectified by doing the volume analysis on event basis. And the seasonal impact on ion concentrations was shown that autumn and summer ion concentrations were greater than spring and compared to winter ion concentrations. These are some of the references, and I thank you very much for your attention.
Many thanks to Tara for your presentation from Germany, and many thanks to all speakers. Let's go to discussion panel. Who want to
so please
ask
switch off
question to our
this
speakers?
room number
Who want
1,
to ask question
because we
to
would like to
Christina
come back to
about
the
your
reality in the
study?
room number 2, because we also are waiting for the discussion. And at the beginning, I would like to again thank you all of you who gave us very interesting talks. With these talks, we had a good beginning of the second day of the conference. And with the discussion, I would like to keep the order of the presentations. So at first, I have a question for you, if you have a question for Marko Spasic. He was the first presenter. Okay.
Yes, thank you. Marko, you showed that the set of species, 3 species, is different from the perspective of soil properties and growth. But for the reclamation of those sites, you showed what is the most important purpose. I would say probably the perspective is to reclaim those sites, and the soil perspective would be more important than tree growth. It's not that this should be forest production areas, I would say. Yeah.
Yes, thank you for your question. I've already had questions, and it's a big sort of debate. What their final purpose should generally be. So yeah, soil is obviously, and in most of the literature, although I do come from like soil science part, just reclamation and returning the functions to soil is one of the main priorities. However, some of the other can sometimes combine more. And it really depends. The mentioned debate about natural succession versus afforestation strategies, and maybe sometimes the assisted succession is also a way to do it. So soil is generally the primary thing, and overall, as you could have seen there, the broadleafs are definitely leading. The alders are completely incomparable to anything else in this sort of region for just soil parts. But there are other things to consider, like biodiversity. Production is obviously not the main function, but there have been some research also from Czech Republic, from Sokolov region, where they compared, for example, aesthetic appeal of certain reclamation sites. And it really, that is also a very important thing, especially for the people living in mining affected areas. So some of the conclusions of those research were, they did like a big Google Documents survey with I don't know how many people, and they asked them different questions, what they think is more pleasing to the eye in terms of reclamation sites. So in most cases, conifer forested stands were better for an average person to look at. There were a few people who have their subjective opinions that like natural succession landscapes are pretty. Personally, I'm not one of those people. Maybe because I do come from some sort of forestry background. I like the forests more and they're a little bit more beautiful. At Antonin Forest Arboretum, for example, there's been some parts which were left for natural succession, and then there are the broadleafs and the conifers tested. Aesthetic appeal, for me, I was amazed, as I said, by the Pizza morica stands, which I've never seen in their like natural habitat, although this is a plantation. But they look really nice, and when you go into like a spruce forest or pine forest, it's a little bit more pretty for the eye of beholder. So yeah, from ecology standpoint, soil is the most important, but also biodiversity is another thing. Over there, the natural succession rules without any questions. And for example, Pizza morica stands were, although they're big, nice, good looking, and from just the tree growing perspective and production potential, pretty amazing. They were rated pretty bad for biodiversity because of the closed canopy, nothing in the ground layers, etc., etc. So it's a little bit in the eye of the beholder, but it's also to the person who makes the strategies what the main goal is. If it's to please the people around, it's to please the people around. If it's biodiversity, then the natural succession is obviously a thing, but maybe some assisted succession or choosing the species for afforestation is really worth considering.
Thank you. One more question, please.
Marco, one question. You showed a study comparing the effect of the age with 30, 50, and 90 years old stands. 30-year-old stand was black alder stand, and those older were Acerxodoplatanus stands. How you can say about the effect of the age when you didn't have the same tree species? It's how you can explain that?
Okay, actually, the fact that I may have forgotten to mention is that all of the three mentioned stands had Alnus glutinosa in the early stages of their development. So it was used as a non-target species to ameliorate the soil initially, and they were usually, according to the law in Czech Republic, cut after 10 or 15 years, leaving just the target species on site. So technically, all of them started with Alnus glutinosa. The youngest site stayed there. The issue in finding sites of such a different age with the same vegetation was an issue. So we tried to do our best to take three broadleaf dominated stands and compare them among themselves. But what we got in the as a main conclusion is that definitely the effect of time is really, really important. And from the other study, which compares the other stuff, you could see the effect of alders. Yeah.
Any other question to Marco? So thank you very much. The second presenter in our session was Karol Tomczak, and I hope you will also have questions to him. Oh yes, please.
No, Karol, very interesting results and also with very practical applications, because if you demonstrated that on former agricultural lands, trees grows very well, and also from the quality of the wood point of view, it's very nice. But I'm wondering if you have also an explanation for your findings. So for you, why you found that in former agricultural lands, trees grow somehow better than on former forests?
Okay, so from this research which I presented, there's, let's say, no explanation, because we didn't check the trees inside, let's say, and yeah, we didn't check the inside, what is inside of the trunk. So we assumed that the initial growth of trees on former farmland is faster, and according to the literature about the birch on former agricultural land, we can see that there is a trend that in the initial state trees grows faster and get bigger dimensions. So that's the explanation. The explanation can be that the accumulation of organic matter of such plots is higher at the beginning. So the trees have better start.
I have also some potential explanation. Isn't it connected with the fact that birch has a very flat root system, and in the post-agricultural lands, the nutrients concentration is the highest in the topmost layer of the soil, and it's well prepared?
Yeah, it can be also the explanation, but we, as a more wood quality focused scientist, we didn't focus on such a matter at that time. Now, when we are cooperating with guys from Czech Republic, we also do soil analysis in such studies, so the future studies will be more complex and we will explain such issues.
Great, thank you. Okay.
Thank you for the responses. Do you have any more questions for Karol Tomczak? If not, we can move to the questions to Cornelia Amon.
Thank you. Based on your research, could you make some recommendations for regional policy or governance about making new stepping stones?
Based on my results, yeah, I would say that it should be balanced, that you have at one hand the preservation of the carbon stock, like I said in my presentation, and then also to look specifically for rare ecosystems and rare habitats to protect them, and also, yeah, just to protect the species that are adapted to these special sites, especially when they're displayed to extreme drought or something, because it will be important for climate change in the future.
Any other question? If not, I would like to ask the question, because one thing that I remember from your presentation is the statement that the protected areas should be chosen wisely. I would like to ask, what is your opinion related to your country, to the Austria? The protected areas are big enough, or they should be changed or enlarged?
Generally, more connected. It would be very good that when we have like more sites that are connected, and also with thinking about the geographical regions, and also the growing regions, like not just randomly connected, but taking into account all those things. Yeah.
Thank you. If there is no more question to Cornelia, we can move to the question to the presenter, Katarzyna Rawlik. Any question? I have a question. Can you explain us what is the reason, or what were the reasons why the decomposition rate was so different between the selected species? Because some of them, the rate of decomposition was very fast, and some of them species presented the very slow decomposition rate. What are the reasons?
Thank you for your question. This is connected with functional traits, like as I said, and in this pool of species, in my opinion, the most important is allocation of biomass to different organs. And spring ephemerals, which decompose the fastest, they are the most of the mass allocate to leaves, and they have no big stems, and these stems are much hard to decompose than leaves, and I think that these species, summer green species of grasses, they have allocated the biomass to stems, which are much hard to decompose. They are more lignificated, and my project is in progress, and I will analyze chemical traits too in the future. So I think it helps to explain it more deeply, the reasons of these differences. Thank you.
Thank you for your answer. One more question.
Kasiu, you showed a very high value of K, 19 something, and as far as I know, this is the highest K value I've ever seen. Did you see somewhere higher value, or did you show us the world record of the decomposition? What is this value on the background of the literature? You know it's better. I'm not the composer, but you know better.
Thank you for your question. I think you may be right, because I studied the same species. It was Ficaria verna, but only on oak Horbin, in oak Horbin forest, so it's not so rich habitats. And this I have the same species on, but the decay rate in oak Horbin forest, it was high too. It was about 14, I think. So now I have Alder forest, which is even rich, and the decomposition is faster. But I think it could be the record of the world in decomposition. So thank you for this suggestion.
Any other question? If not, I think we can finish our session. It was a pleasure to lead this session. So thank you again for your presentation. Thank you for your answers and discussion. And now we have the coffee break. So we can leave this room and drink something, eat something, and wait for another session. Thank you very much.
Hello, good morning and welcome everyone on this place. My name is Tomasz Leski and I have a pleasure of leading this session of our conference. In this part of our conference, in this session, we have 6 presentations on the agenda. Since there are 2 parallel sessions in this side room and another, it is very important to maintaining the time of regime. I would like to remind you that each speaker have only 15 minutes. According to the conference program, the time for the discussion, questions and comments will be is planned to on the end of the session. So we may start. The first presentation by Marcel from Brazil about the ecophysiology of 2 woody plant species. The floor is yours.
Thank you for introduction. So, hello. My name is Marcel. I am PhD student in Brazil, and I will present for you some data from my master thesis, don't publish yet. And I will talk about temporal modulation of ecophysiology of Schinus cerambytifolia and Alder castophilum used in revegetation of a fragment of the Atlantic forest. So, tropical forests are home to more than 50% of planet's biodiversity, and these environments are considered the richest in this regard. So, looking to this picture, we can see in the dark green the tropical forests around the world. In Brazil, we have 2 types of tropical forests. The Amazon, located here in the north of the country, and 60% of the Amazon belong to Brazil. And the main challenge to conserve this area is stop the deforestation. But I will talk about other type of tropical forests, the Atlantic forests, located in the coast of Brazil, where live 80% of the population in the country. And these tropical forests have been explored a long time, by centuries, so rest almost 10% little fragments spread to the Brazilian coast. So the main challenge here is recover these areas. So, recovering degraded areas by years of exploration require revegetation projects. However, plant seedlings is not a simple task. Well, in this perspective, the object of this work was investigated the effects of different soil treatments associated with the presence and absence of a type of green manure on the ecophysiology of 2 species native to the Atlantic forest, Alder castophilum and Schinus cerambytifolia. So, here we can see an area where the experiments were conducted. Here in this area this area was used to clay exploration to put the machines in each spot here to oil exploration. So this is a area rich in petroleum. So today in 2024, we can see this point where the experiment was conducted and the experiment. I think so. It's okay. So we tested 3 soil treatments, because the traffic with the heavy machines here compact the soil a lot. So we must discompact the soils. So we test 3 treatments: scarification, scarification plus herring, and subsoiling plus herring. And we also test a type of green manure to improve the seedling survival in this environment. And this green manure is compound a mix of seeds with leguminous, cruciferous, and forage species used as a green manure in the revegetation experiment. Basically, we use native species of beans to fix nitrogen in this area. And our plant material was 2 native species called Schinus terebintifolia, belong to an Arcadiaceae family, and Dalbergia castophyllum belong to Fabaceae family. So this is a non-nitrogen fixer species, and this is a fixed nitrogen species. And we collect some data, fluorescence, and collect the leaves to analyze the photosynthetic pigments content in laboratory. And we collect some growth traits like plant height and stem diameter, and perform principal component analysis using our program. So the first result is this principal component analysis, and when all the results as a function of species. So we can see 2 clusters. The orange one is the Schinus species, and we can see the data correlated with the chlorophyll content and the photosynthesis parameters. On the other side, we can see the red one cluster is Dalbergia castophyllum, correlated with the carotenoid content and growth traits. When you perform this analysis as a function of the time, we can see a difference over the time. At 6 months, the data collect is correlated with the chlorophyll content and photosynthesis parameter. At 12 months, the data are correlated with the carotenoid content and growth traits. Okay, but this is a good or a bad thing for our seedlings here. So we perform other statistical analysis to prove it. So to Dalbergia castophyllum, we have at 6 months, and we don't have a clear tendency here in the carotenoid content, and are reduced in the chlorophyll content in plants with green manure. So, this is bad news. But at 12 months, we have a clear tendency also to carotenoids content for the chlorophyll content, and in all soil treatments, seedlings growing with the green manure showed more carotenoid contents and chlorophyll contents over the time. So this is good because carotenoid made part of chlorophyll photosynthesis process and acting against some stress in the environment, and chlorophyll is directly correlated to the photosynthesis process. Well, 2 second species, a non-fixed species, Schinus, we have show the more carotenoid contents in 2 treatments at 6 months, and the same behavior we observe in the Dalbergia. And at 6 months, chlorophyll content was lesser here and with the green manure. But at 12 months, we can see the same behavior. The carotenoid content was higher in plants planted with green manure in all treatments, and just for the subsoil plus herring treatment in chlorophyll content. Well, so this changing in the pigments content affects the photosynthesis process. So the photosynthesis process increase in plants with green manure in all treatments for both species at 12 months. And these parameters, SFI-ABS, refers to structural and functional events in photosystem 2 that lead to electron transport in photosynthesis. And PABS refers to PS2 perform index. So when we use the green manure, we change the photosynthetic pigments, and this affect the photosynthesis process. So, in summary, plants growing without green manure show higher levels of M0. This reflects closure by central reaction in photosystems 2 here, and this is not good to the photosynthesis process, and reduce the efficiency of the photosynthesis. So when the plants grow up with the green manure in all treatments, the central reactions keep open, and this impact in the efficiency of the photosynthesis process. So this also reflects in the growth rates. So for Schinus, a non-nitrogen fixing species, the treatments, the plant height and stem diameter only differ between soil treatments. And we can see here at 6 and 12 months, plant height at 6 and 12 months stem diameter, subsoil plus harrowing will the best soil treatment to grow of this plant. And to the Albergia a nitrogen fixed species, the plant height and stem diameter differ between the manure treatments at 6 months here and at 12 months in stem diameter here, and also differ between the soil treatments, show the highest values and also in subsoil plus harrowing treatment. So green manure affect positively over the time the physiology and growth of both species plants. Thus, the subsoil plus harrowing technique that promote the highest degree of the soil decompactation associated with the green manure demonstrates its potential used in projects of revegetation of degraded areas. And we can use the same machines that prepare the soil for plant crops to plant trees and revegetation projects in a fragment of Atlantic forest. So thank you for your attention.
Thank you for your interesting and clear presentation and keeping the time. The next point in our agenda is the presentation of Marta Górska from Poznań Life Science University. The floor is yours.
Can I adjust? So I'm the first person to say good afternoon. It should be okay. I will present today the part of the research on the effect of cement dust on trees. And the story began in the North Estonia, where the Kunda cement plant is located. It emitted a heavy amount of cement dust over almost 30 years, since 1960 until 1996, when the filter were assembled in the factory. And the dust deposition was highly alkaline. So we already knew that it caused the soil alkalization in the area of Kunda cement plant, but we wanted to know how much of the element contained in the cement dust deposition was absorbed by the trees growing there. So we selected two sites, one in the close vicinity of the Kunda factory, and the second one in the National Park Lahema, which was relatively not affected by the cement deposition, the cement dust deposition. And in the area marked by one here in the close to the factory, the dust deposition reached up to 2.5 kgs per square meter. So you can imagine per year how polluted was the area. And we selected 20 trees from each plots, 8 years old Scots pines, and from each trees we bored 2 increment cores, and also selected some bark and soil around the trees. Then we compared them. Between Lahema and Kunda. At first, we analyzed the soil texture, and in the result, we saw that grains from Kunda area were slightly finer than in the Lahema area, and that alkalization of the Kunda soil was higher, and also the soil electrical conductivity was higher, which indicated the salinity of the soil. So it remains higher after almost 40 years since the emission stopped. In the next step, we mineralized all the samples to enable them to analyze in an ICP spectrometer. And we analyzed 14 elements content, from which 7 of them were abundant in the dust deposition, and they are marked in red on the left side. We compared the concentration using T-student test, and as we could expect in the soil from Kunda, the concentration of the elements, which were the main component in the dust deposition, was significantly higher. The only exception is for potassium, which was higher but not statistically significant level. And we also, if it comes to the wood samples, we split them into 2 parts. The outermost, which were produced or grown after the dust deposition stopped, and the more inner parts, which were created by tree during the deposition time. In the inner part, we found out that 3 elements were in a higher concentration in wood from Kunda. It was calcium, potassium, and magnesium, and 3 of them were the main components of the dust from the cement plant. Almost the same pattern were observed for the wood from the outer parts. Only the chromium joined the significant differences between the locations. And we also made a comparison between the wood from the inner part and the outer part for the Kunda location. And we found out that for calcium and magnesium, the inner parts were richer in those 2 elements. But the opposite effect we observed for potassium and phosphorus, which are though mobile in the plant. Additionally, we calculated the bioconcentration factor to find out how effective was the absorption of the elements relatively. I mean, comparing to the amount of the elements contained in the soil. And we found out that the BCF indexes were in general higher for the control area, which shows some limitation in the absorption or uptake of the elements in any area. Unfortunately, the volume of the bark samples did not allow us to make some enough replication, so we could not perform the statistical analysis. But the general view on the data shows us that in Lahema bark there are generally more elements. The only exception is the calcium, which were abundant in Kunda, as you remember. So even after 30 years since the pollution stopped, we still observe the soil more alkaline in Kunda and more rich in the elements contained in the dust deposition. And also in wood from Kunda, from trees growing in Kunda, are richer in those components from the dust pollution. Although the bioconcentration level is higher in the Lahema Park. Thank you for your attention.
Thanks a lot for your clear presentation. And without delay, let's continue to the next presentation by Marta Kujawska from our institute and from my lab about ulmus levis and microbiome.
I'm Marta Kujawska. I'm working in the Institute of Dendrology. It's a really big pleasure to talk to you today. And that I said, Professor Leski, tomorrow, Professor Leski and I are waiting for you in our lab. And I hope that we have a lot of fun with working with mycorrhizas. Today, I would like to say more about mycorrhiza, but invisible mycorrhiza. Yesterday, you could hear that the trees have no roots, but have mycorrhizas. And today, I give you the reason why the mycorrhiza are fun. Why ulmus levis? Ulmus levis is one of the three species elms native to Poland, very largely distributed. And ulmus levis generally the best persist, the best survived after Dutch elm disease. I hope that you know what was the Dutch elm disease and the devastation and severity of its pandemic focused the interest of researchers of generally the limitation of this DAD and other aspects of this disease was significantly neglected. Of course, the topic of mycorrhiza also. So generally, the elms are truly forgotten for a long time. Fortunately, in the recent times, we can see that elms are in the better condition, and so this is the reason why the ulmus levis is the topic of my research. History of research on elm mycorrhiza, it's not easy. The knowledge on the mycorrhizal symbiosis of elms is poorly recognized. The first report 100 years ago about mycorrhiza of elms pointed only the occurrence of arbuscular fungi, invisible fungi inside the roots of trees. And in this time, that was named as endomycorrhizas. However, Klecka, Volkov, and Meyer mentioned that elm trees can form also ectomycorrhizal symbiosis. Ectomycorrhizas are observed perhaps on roots in elms on this time. And what is interesting, that also on the list of the type of mycorrhiza in Great Britain trees, they also mentioned that elms can form this two type of mycorrhizas. However, Brandon and Teders, a very, very important person in mycorrhizal world, suggested that elms cannot form ectomycorrhizal symbiosis. Why? Because elms have beaded roots. This type of roots are very difficult to observation and to clear and to stain. They have a lot of lignin, a lot of tannin, and probably the results that indicated that elms have ectomycorrhizal symbiosis was based on the observation of sporocarps that grow nearly the elm trees. This is not true, and so we think about check this compilation, this problem. What is exactly in these roots, in elm roots, and maybe we can solution this problem. Of course, environmental factors strongly affected the dominance of the type of mycorrhizas. The trees can switching arbuscular or ectomycorrhiza type depend on environmental environments, and of course other factors like tree age, season, tree health, habitat type also influence. So today I would like to focus on only a few factors: habitat type and season, and a little bit tree age. Yes, methods, basic morphological observation, microscopical observation that I said before, that was really horrible and not easy. Metagenomic analysis based on ITS 2 region, and of course base molecular analysis, Sanger sequencing to check that ectomycorrhizal fungi form this symbiosis or not on the elm roots. I would like to see the pictures, very characteristic elements of arbuscular fungi, like coils, arbuscles. Yes, here we coils, like sempertine, and see on the arbuscles on the little trees inside the roots. Very beautiful, but it's not easy to see on this type of roots with a lot of lignin. And to our study, we choose three main type of environments, and forest habitats, including riparian and mixed forests, and non-forest habitats in urban rural and post-industrial areas, and forest nurseries. In each type of environments, we chosen three types, and we sampled the soil with roots of elms into sampling periods, spring and autumn. At the beginning, the results of observation from microscope. As you can see, the share of total characteristic typical arbuscular structures is the highest in forest nurseries and on the young seedlings of elms, and the lowest in urban and post-industrial areas. Also, the share of non-mycorrhizal roots, so without elements of mycorrhizas, was observed also on disturbed areas. And now we're moving on metagenomic analysis. The mean species richness was the lowest in forest nurseries and the highest in forest habitats. So the reason why it's the lowest in forest nurseries is easy, because the young seedlings can have less the partners, fungal partners, than the other trees in the natural habitats. And very similar situation in case of Shannon diversity. Here we have the share in the soil fungal communities between each trophic groups, and as you can see, the share of symbiotic fungi is higher in forest habitats than non-forest habitats, and in rural areas is the most varied the share of each type of trophic groups. And now something, a little word about composition of fungi in each type of habitats. It's very similar. Why? The non-specific fungi constitute in each type of habitat almost 15–20%, and the most abundant fungal family constitute 60–70%. Here is a situation in most mixed forest and riparian forest. The same, very, very similar situation in urban areas, but here the percent of non-specific fungi is raised, and this more varied situation, more varied fungal composition in rural areas, what we saw in earlier pictures. Post-industrial areas also, the non-specific fungi higher the amount, and forest nurseries. So two enzyme analysis showed that type of habitat is the most important factor that influence that shape this composition, and season not. So this is the summary and conclusion, the first part of my my research. So the type of habitats affects the structure and season not. Forest nurseries are characterized by the higher share of arbuscular structures. Maybe surprising at the beginning, but when we think about why the young seedlings need more nutrient, they grow all the time, so the amount of the structure is higher than in the case of other trees. And the second part of my research project was pot experiment. We think about what neighborhood trees in seedlings in pot can influence the type of mycorrhizal colonization of elm seedlings. So we have 8 variants. First variant was only seedlings Ulmus levis. The second variant was Ulmus levis and maple. So this is also characteristic tree species which can form only arbuscular symbiosis. And the next seedlings, Carpinus betulus and Quercus robus, form typical ectomycorrhizal symbiosis. So this is the mixed arbuscular and ectomycorrhizal partners in one pot. And microscopic observation, the highest share of total arbuscular forms are in variant 7, where we have oak, seedling, elm seedling, and maple. And from soil samples, from metagenomic analysis, we also see that we didn't have significant differences, but the lowest number of otus, fungal otus, was also in variant 7 and in variant 3. And very similar situation in the case of Shannon diversity. And in the case of relative abundance of soil fungi from different trophic groups, the most important conclusion for this picture is that in variant first and second, we didn't have symbiotic fungi. Why? Because the ITS2 region and this pair of primers using in this metagenomic analysis is focusing on ectomycorrhizal fungi. And in variant first and second, we have trees that perhaps form symbiosis only with ectomycorrhizal, only with arbuscular fungi. So maybe this is the reason why this share of symbiotic fungi is very, very low here. And the NMDs, two-way analysis show also that the control variant first and second is different in the case of fungal composition than in the other variants that include a mixed arbuscular and ectomycorrhizal symbiotic partners. Okay, conclusions. So the neighborhood definitely influence to composition, and we hope that if we add our results from soil chemical analysis and root fungal communities from not soil samples but from root samples, we have the whole picture of microbiome, soil and root microbiome of elm trees. Thank you for your attention.
Thank you, Marta, for your rich presentation. And the next speaker is Birch from Slovenian Forestry Institute in Ljubljana.
Thank you very much, Chair. Okay, hopefully this works and you can hear me. 1 second. So I'll be talking about adapting artificial forest regeneration to climate change. And I will be showing you some perspective from Slovenia and our team, also with my professor, Dr. Hojka Krajger. I'm just in the first year of my PhD, even though I'm not the youngest. So we will be talking more about some concepts, some data on Slovenian forests, and not so much about scientific data. And then I will also present some ongoing projects that we are doing at this moment. So Slovenia is a very forested country, a forest nation. Down there you can see the map. So it looks very green, and we are very proud about our forests. We have a rich history of sustainable forest management, which goes back to, let's say, 1771 with our first forest management plan with the principle of sustainability, which we celebrated a few years ago, 250 years of this forest management plan since it was created. In 1892, the first forest reserve in Reichenauski Rog. And then we go to the Slovenian Forestry School after the Second World War, and we achieve almost 50% of forest in Natura 2000. So our main pillars are sustainable, close to nature, and multi-purpose forest management. So it means keeping all forest functions for future generations. So we are giving some functions or ecosystem services a priority, but we are still keeping the rest. So how it looks in practice, you can see mainly this data. So you can see that some key metrics went up 3 times, basically, the growing stock and so on after the Second World War. So economy and nature conservation can go hand in hand, in our opinion. So here, I won't be very long. Let's just look at some data. So our main 3 species are beech and Norway spruce and some silver fir and oaks. We have 1 million hectares of forests, some very good biodiversity indicators, and 80% private forests. So every 5th Slovenian is a forest owner. So you see that this close to nature management system was very successful in the past. But what has been going on since 2014? We had this sleet. In February 2014, we had 10 centimeters of ice on all trees, cars, you know, and it had horrible consequences. You can see how many forests were affected, great damages for us, great regeneration projects, big ones that weren't present before. So this is 10 years ago. This happens. This is the before that there wasn't such an event in forest. It's not recorded. So this is the map of sleet, and then you have bark beetles and windrows. So a lot of forests were damaged, especially spruce, which was planted artificially too low, you know, at too low heights, altitudes. So then we have a few years of peace, 2 or 3, and then in 2022 the forest fire comes to Karst, damaging 3,000 hectares of forest. It was catastrophic. And then in 2023, it wasn't so bad for the forest, but for the country, we had extreme floods. So apparently, you know, in the last 10 years, something is happening. Something very bad is happening. Something is becoming different. So if we look at the data, our climate predictions, we are now here. So something is happening for the last 10 years. And this is, you know, the good scenario. This is the basically most probable one, and this is the bad one. So we are going even further in this direction. The seasons are shifting. We have late frost now, almost each year. We have most common extreme weather events, as I showed you. We have sunnier, hotter, and drier summers, but this is not a rule. Some summers are wet. And climate change is definitely affecting forests. So this is just one map from Hanewinkel from 2012. It's an illustration that something is happening. Probably the forest will change, like we heard many times in this conference. And this is for Slovenia. So this is the high scenario. So basically, you see here you have a lot of mountain forest, temperate forest, and it, in the worst case, the growing conditions, climate, it goes basically all to Mediterranean. You know, this is, I think, in 70 years. I'm not sure, but It's quite so. Okay, we spoke about close to nature forestry and how successful it was. This is very important, is that we have 90 to 95% natural regeneration. So what can you do here in terms of planting? So even though it's natural, it doesn't mean letting go. We are still foresters. We still need to help forests adapt to climate change because climate change is human-induced. So these are our goals, our sustainability of forest functions or ecosystem services for future generations. And then we have biodiversity conservation and climate change mitigation, so basically carbon capture, etc. So some principles would be the proactive forest management, increasing diversity. This means spreading risk, so also in forest structure. You know, we need more young forests. We have quite, not the best structure. We need more young forests. And we need to increase, if possible, of course, in a natural way, species and genetic diversity of forests. So this is the key. We don't want to make a map, a decision, and say this is how it's going to be and plant this genotype. But we need to conserve the adaptive potential of forests. Maybe, change the mix of tree species. The spruce monocultures, this is especially we need to convert and maybe even faster. Lower timber stock as risk management. Also find clones that would be resistant to drought and maybe add some of that to the mixture. And this we would do by planting these different trees into natural forests in maybe a smaller scale, and really look at this assisted gene flow to help populations adapt. So I really like this graphic by Sylvia Schuller and colleagues from Vienna. So these are basically our lines of defense against climate change. The use of artificial regeneration in close to nature forest management can serve as risk mitigation insurance policy for us. So what are we doing in our group at our institute? So we are making provenance trials. We are going to make an assisted gene flow model, assisted migration model for transfer of provenances. We are looking at new technologies to enhance survival of seedlings when planted. Strategies for monitoring, this is also very important because you can plant, but it's also very important to monitor what is happening so you can do it better in 10 years. So we are doing a lot with stakeholder cooperation, policy and business models, and we are building a new center, which I will show you later. This project is wrapping up. It's called Life Systemic. It was, I think, a 4-year life project. Its aim is to use the tool of genetic diversity to best protect forest against the impact of climate change. It's led by University of Florence. There will be, now we are in the final stages, so there will be several manuals. There will be also a model called Gen BioSylvie. You could also maybe take a look at. And it concludes very soon. We have also a special section of this project at the UFRO in Stockholm. So you are welcome to this session. It's Monday, 24th of June at 4 o'clock. So our previous project was Live GenMon. Its main output was the manual of forest genetic monitoring. So if this is something you would like to look at, it's free to download. And we are also part of these big European projects of the Horizon mechanism. So I don't know if you heard about the My Garden of Trees. It's a project from Switzerland, from VSL, led by Dr. Katalin Chillery. So the aim is to make small provenance trials with citizen science. So they gather the seed and they sent it to volunteer foresters. And we are just helping in Slovenia with this project. So we have 25 of those micro gardens, I think with 20 plus or 30 plus provenances growing. So this will be also some very nice data for, let's say, assisted migration we'll see what is working in a few years. So we're also having at our premises a macro garden, which will be a bit larger, and my garden of trees is just silver fir and European beech, I forgot to mention. So this is the next thing we have started this year, so try to increase the success of reforestation by planting after the karst forest fire with hydrogel and mycorrhiza. So, so this is the hydrogel, and then it takes up a lot of water, so it becomes such a big bulb of water, and it helps seedling. It should help survive the drought. The seedlings. So this is the karst. So here it's very dry, it's very rocky, you can see. So you have to look for pockets where to plant. And this is just mycorrhiza on bare root seedlings. So you just dip it into the mixture. So this is a very practical thing. We'll see if it works. It's just something we tried in cooperation with the local foresters from the Slovenian Forest Service. So, and here, because you can't plant in rows, we also used precise GPS to monitor the seedlings. So this could be very nice because in 30 years we can look at which seedlings survived, but they are also marked with this. So then you can make a really nice map of all the seedlings in GIS. We are also working with digitalization in this project, e-forestry. So we are looking to create a digital national register of forest genetic resources, ensure traceability and use of forest reproductive material, provision and use, help with the logistics with this system, and improve the monitoring of fruiting of forest trees and so on. So then we really know when they are masting, because this is becoming a big problem for a seed provision. So this is the new Center for Forest Seeds, Nurseries and Forest Protection. It hasn't started to be built, but it will start very soon. So we will have at our premises a new center, almost 3,000 square meters of research space for forest seed production, dealing with all the topics we mentioned before. We also do some policy work. I won't go through this, but we also provide services to business and policymakers in some analysis. This is also very important, maybe more than some other things, but it's the last is the forest pedagogy. So we also take some time to host children and visit schools. It's very important because at least in Slovenia, forestry is losing a lot of its place in schools. Their children don't prefer, let's say, proactive forest management. They are taught more to conservation. So this could be a problem in the future. And you can download this handbook also. You have activities there you can do yourself. Okay, so yeah, I'm also a part of the team of the this UNEC FAO Forest Communicators Network, and you are welcome to our workshop meeting in Vienna in December. Thank you.
Thank you, Boris, for your interesting story about Slovenian forestry and activity. The next, once again, the best wishes for Hojka. Then let's move to the next presentation by Sebastian Burre from our institute. The stage is here.
Hello everyone, my name is Sebastian Burre and I'm a PhD student from Department of Ecology from Institute of Dendrology of the Polish Academy of Sciences in Kórnik. And today I want to present part of my PhD studies about the impact of Prunus serotina on natural forest regeneration. And at the beginning, a short introduction about this species because maybe someone are not familiar with this species. So Prunus sterotina, black cherry, naturally occurs in eastern part of North America and was introduced in Europe in 17th century. Then in 19th and 20th century was planted on a larger scale in all Europe, and foresters believed that it will be a very good productive species and also phytomeliorative. Unfortunately, now Prunus serotina is one of the most common species, invasive species in Poland and in Central Europe, and are used mostly as a fuel wood. And unfortunately, Prunus serotina can affect negatively on biodiversity. And also, in some places in Poland and in all Europe, foresters must eradicate this species before artificial regeneration. And first of all, natural regeneration is key element of forest sustainability, especially in the close to nature forestry. And we should take into account all variables that can affect this natural regeneration. And we aim to study the impact of Prunus serotina on natural forest regeneration, and we are focused mostly on the scale effect or per capita effect of Prunus serotina. And we hope that our studies will increase our knowledge about invasive species and increase management of invasive species in Poland. We hypothesize that Prunus serotina will impact natural forest regeneration along its biomass gradient. And we suppose that it will be a different impact on nutrient-poor sites and nutrient-rich sites. And also we hypothesize that the other alien species will benefit from Prunus serotina presence, according to the invasional meltdown hypothesis. So we established 96 plots, 96 circular plots with area of 500 square meters in managed forests in Poland. In 5 forest inspectorates, it was Babki, Czerniejewo, Jarocin, Konstantynowo, and Łopuchówko. We are now somewhere here, and we are searching 8 replication, including for each of the variants, and these variants include 3 characteristics, which is habitat type, invasion level, and stand age. For habitat types, we distinguish nutrient-rich habitats with oaks, and it was mainly Galliocarpinetum communities or secondary oak forests, and nutrient-poor with pines, and it was mostly Leucobryopinetum communities or secondary forests with pines. For invasion level, we have 3 categories: control plots with 0 coverage, medium plots with coverage up to 30%, and with high coverage more than 50%. And for stand age, we have medium stand and mature stands. To better describe this quantitative gradient, we measure all diameter at breast height for all individuals of Prunus serotina. And then we use allometric models to estimate the above ground biomass, which include leaves, stem, and branches. Okay, on 4 circular subplots, on each plots, we inventorized the natural regeneration. This radius subplots have 3 meters and was schematic distributed. And we distinguish 3 height age classes. First was seedlings, the second was sublings up to half meters, And third one was saplings from 0.5 meters to 1.3 meters. All analyses were made in RStudio, and firstly we use canonical correspondence analysis, CCA, and we used above ground biomass of Prunus serotina, age and Prunus serotina present or absence as constraints. We also used threshold indicator taxa analysis, Titan 2 package, to show that biomass can impact the natural forest regeneration. And finally, we used generalized linear and mixed effect models. And as dependent variables, we use a number of individuals, the species that occurs 20% on the plots. Some of the all native trees and all alien species, but without Prunus serotina. As fixed effect, we used Prunus serotina above ground biomass and age. And as random effects, forest inspectorates and year because the investigation was made in the 3 years, so we must use it as the random effect. Okay, so let's move to the results. So for the nutrient poor habitats with Scots pines, the lowest diameter outburst height was 0.1 centimeters of Prunus serotina, and the maximum was 33. And for a nutrient-rich habitat, it was also from 0.1 to 31.4 centimeters. And if we talk about biomass, for nutrient-poor habitats, the biomass varied from 0.9 to 15%. But I have to say that we also have one plot with biomass of 47 tons per hectare, but we excluded in the next analysis because it was far more than the others. And for nutrient-rich habitats with oaks, the biomass ranged from 0.4 to 27 tons per hectare. Okay, so if we talk about natural regeneration, we inventorized 56 woody species, including 11 alien species. And the second class of the natural regeneration was most frequent. It was more than 80%. First class shared 8.5%, and the third class 10.7%. Okay, let's move to the CCA analysis. So both nutrient poor and nutrient-rich sites depends on the biomass of the species composition of the on this variance varied on the biomass, but the other factors are not significant. Okay, Titan 2 package diverse our taxa on the increasers and the decreasers, and the decreasers has gray colors, and in our case it was only Quercus petraea, and increasers was on the red, and it was Prunus serotina. Because this package, this function is very strict, and purity and reliability of the data must be more than 95%. So the other species are not on the graphs. Okay, and finally we are going to the most important parts, the linear models. So I want to say at the beginning that only Prunus serotina are statistically significant if we talk about per capita effect, but the biggest effect size was in the case of the second classes, because as you can see, for control plots, we have 8 individuals. For the 6 tons per hectare, we have 76, and for the 12 tons per hectare, we have more than 100 individuals. So the effect sizes are very high. The other species is Pinus sylvestris, and Pinus sylvestris decreased with the biomass of Prunus serotina. And it was very important result because on the nutrient poor sites, Pinus sylvestris is the most important species. So they are, and it is in the decreased. The other important species was Quercuses. Quercus petra seedlings are increasing of the biomass of Prunus serotina, but the second classes of Quercus petra are in the decreased, and as well as Quercus robur. And the other Sorbus aucuparia and all alien species are in the increased if we talk about the second class. Let's move to the nutrient-rich sites. Very interesting result we have with Acer pseudoplatanus, because the seedlings are in the decreased and third classes are in the increased. Quercus petra was in the decreased. Carpinus vetellus, what is also interesting because it is very species which are very which was characterized by very big plasticity. And Ulmus minor and Quercus robur are in the increased, as well as Prunus padus third class. Okay, so to conclude, so we state that per capita effect of Prunus serotina are very important, and we should study this. But particular species impact response differently on the Prunus serotina presence. And we can also say about climate changes, because as I said previously, for example, Prunus serotina was harmed by climate changes, and Prunus serotina can empower these negative changes. More important conclusion is about Quercus petra, for example, because we hope that it can be a good species we talk about climate changes, but Prunus serotina can impact negatively or positively, so it's very interesting. And finally, our results are very important for the Prunus serotina management in protected and managed forests. This study was financed by National Science Center. And special thanks to Martin Dyderski for our support. Thank you so much.
Thank you, Sebastian, for your nice presentation. Now we've come to the final presentation on our agenda. The last speaker is Pravin Dashti from Iran.
Hello everyone, my name is Pravin Dashti. I hold PhD graduated from University of Khorasan of Iran. These studies is about it's part of my thesis and we analyze the import demand function of Iranian paper and paperboard using band test. Global trends in the forest products industry since the 21st century have seen increased demand in developing regions and decreased consumption in advanced countries. The demand for wooden products in Iran is increased, and imports are considered in pivotal factors in regulating the wood market. Importing paper face challenges such as currency depreciation and economic sanction, promoting the need for strategic attention to market dynamic and policy formulation. Sanction imposed following Iran's nuclear program in 2006 include cutting bank ties with the global network and implementing oil sanction. This sanction had a significant economic impacts, affecting cross-domestic products as well as imports and exports. Nevertheless, their impact on paper and paperboard import demand has not yet been truly examined. The main objective of this research include the following: determining the long-term and short-term import demand function for paper, investigating the impact of macroeconomic variable on import, assessing the effect of economic sanction on the paper import market, estimating the long-term and short-term elasticity of variable under investigation, in estimating the error correction model ECM to determine the speed of adjustment from the short-term model to the long-term equilibrium. Material and methods. In this study, we examined the trend of wood production of Iran, the trend of paper and paperboard imports, estimating the demand function for paper and paperboard. The data used for this study include time series data and include import quantities of variable types of paper, domestic wood production, import tariffs, exchange rates, gross domestic products, GDP excluding oil, and oil revenue and relative price. Time period was 1984 to 2017, about 34 years. The data were collected from this source: FAO, OPEC, Forest Range and Watershed Management Organization of Iran, Foreign Trade Yearbook and Central Bank of Iran, Custom Administration, National Portal of Statistics, Iran Industries Information Bank. This equation represented the import demand of function. We say the variables such as imports of paper and paperboard, GDP, relative price, exchange rate, tariff, average applied tariff rate on imports, oil revenue, domestic wood product, and dummy variable reflecting the sanction during the specific years. We use the ARDL model and band test for achieve these goals. In the first stage, all of variables converting to the logarithm form, because logarithm form give us the elasticity directly, and then stability of variables using the argument Dickey-Fuller and Phillips-Brown test examining. In the second stage, we estimate the dynamic equation and coefficients of short-term model. The third stage, the existence of long relationship among the variable is examined, and it found error correction model or ECM is estimated. At the end, coefficient stability and model accuracy are examined. In result, timber production from Iran forests experienced an increase from the early 1916 to mid-1970, followed by the decrease you see in this chart. And two charts indicate that the increasing trends of paper and paperboard and pulpwood in time study. The result of short-term and long-term model and coefficients you see in the first table. The result of the second table indicate the existence of long-term relationship between the variables. GDP, the study found that GDP growth, both short-term and long-term, had the greatest impact on paper imports. Increased national income led to higher demand and consequently increased paper imports. Domestic wood production from north of Iran, the just forest, chemical forest in Iran, had a negative impact on paper imports, with pull factors of minus 0.7% and minus 0.6% in the short and long term, respectively. Decreased domestic wood production has led to increased dependence on imports. And economic sanction, a new variable in the study, led to decreased paper imports. Short-term reduction coefficient is minus 0.7%, 5%, and long-term reduction coefficient is minus 0.41%. Sanction caused difficulties in purchasing and financial transaction, indirectly impacting the decrease in GDP. Exchange rate. The depreciation of national currency led to increased cost for paper imports, consequently causing the decline in demand. Relative price. A 1% increase in the relative price of imported paper has resulted in a 0.3% decrease in imports. Tariff rate and oil revenue had not significant effect on imports. The government tariff rate, the government imposed the low tariff and value-added tax exemption on certain paper products, rendering them ineffective. And oil revenue, the revenue obtained from oil exports, did not have a significant impact on level of imports. Error correction model, or ECM, show the speed of adjustment. Error correction coefficient is minus 1.26%, implies rapid equilibrium adjustment, typically within one period. You see some reference, and thank you for your attention.
Thank you for your presentation. And once again, I would like to thank all speakers for their presentation. If you are ready, we have time now for ask questions, comments, discussion. Now I open the floor for discussion and questions. I suggest starting with questions from the last presentation and the next ones. Who's first?
I can have one question. Do you maybe know why did the timber production lower so much? The chart. Why did the timber production go down?
Project in Iran that forbidden harvest from forest for 10 years. For 10 years, because rest of forest and recovery forest is main resource to reduce timber production of Iran, I think.
you. Any questions for Erwin? If not, we may move to the Sebastian presentation about Prunus serotina. Do you have any questions for Sebastian? Silence. Yes, Sonia.
So just a general question maybe. Can you tell us about your future plans? What's next about it?
Okay, maybe I say that also in my PhD studies I was also study different species, which is Robinia pseudoacacia. So we will see maybe in the case of Robinia pseudoacacia the results will be totally different because as we know Robinia pseudoacacia has much more sizes, bigger sizes, and its nutrient is oh my God! Yeah, in general nutrient, but nitrogen cycling was totally different because yeah, she can live with special bacteria. And if we talk about Prunus serotina, I was thinking because, as I said, the second class, the share of the second class was more than 80%. So maybe in the publication it will be better, for example, to summarize the second class with the third one because it was only 10%. So more maybe the results will be better. Yeah, but I will see. Thank you so much.
Okay, any other questions for Sebastian? Still science. Okay, we may move to the green country, Slovenia. Yeah? Okay, to the Sebastian.
Could you maybe elaborate a little bit about on the ways to eradicate produce from the stands? I mean, we've heard yesterday, yeah, in the talk that it would be good to keep closed canopy, maybe to prevent its spread. How do you think about that? What do you think about that?
Okay, if we talk about Prunus serotina eradication or assimilation, in the Netherlands, there are a lot of very important studies of Nissen, and he thought that in the case of nutrient-poor sites, for example, when we have to planted the pines, so we must remove the Prunus serotina because it's very difficult to produce trees, yeah, from the seedlings or saplings. And if we talk about nutrient-rich sites, the assimilation of Prunus serotina was easier because there are a lot of understory of native trees, so there are concurrency for Prunus serotina. But to compare with my results, in the nutrient-rich sites, Quercus petraea and Quercus robur was very important. So maybe we should also eradicate, yeah? If we would have a regular generation of oaks, so maybe we should support it and reduce the quantity of Prunus serotina. Yeah, so it's not straightforward. Yeah.
Thank you. Any other questions about Prunus? No. Once again, move to the Slovenian green country.
Thank you, Boris, for a very nice introduction to Slovenian forestry and overview of what you are doing there. So my question is maybe a comment or a question. Because you said that the sustainability is the priority regardless of ownership. So private owners are also, what I understand, are also forced to use the natural regeneration, and they just can't do what they want. So it would be difficult in this setup to come with some solution to what you are looking for, like underplanting with some pre-adapted populations or genotypes for assisted migration. So I'm just trying to think aloud. Maybe what could you do? Because this adaptation to climate change is important from your standpoint, and of course it's important all over the world, and we should incorporate that in forest management decisions. Just my thought would be maybe to use the option is so-called what was it called? I just forgot. Composite provenancing, yeah, composite provenancing. So mixing maybe some seed sources from pre-adapted from the areas with the climate, which would have climate projected for the future at the other sides. So maybe trying to move seed sources from lower elevations to some upper elevations. In your conditions, you have a lot of variability, the altitudinal variability also. So, yeah.
Thank you very much for this comment. It's very valuable. Forest management is done according to forest management plans, which are prepared by the State Forestry Service for all forests. So basically the question is, how sure are we that we are right? And when we are sure, this shouldn't be too difficult to put into practice. So, but I think we should start with slow steps, like you said, with maybe mixing a little bit. Thanks.
Okay, Boris, you stated that we have to help forests to adapt to climate change, as climate change is anthropogenic. I wanted to ask if you think that forests cannot self-regenerate.
Of course they can. It's all about, again, humans and sustainability of forest functions. So, of course, forests will be there, but will they continuously provide all the forest functions or ecosystem services that society is looking for from forests? So this is then the need to adapt or, make artificial regeneration. Otherwise, in this climate, the forests will grow.
Thank you very much for your presentation. I have one question regarding the bark beetle infestation. You mentioned this is one of the risks in Slovenian forests, especially in spruce. I did not understand the connection between how climate change is influencing this infestation.
Yes, this is the problem, I think, for most Central Europe. So there was artificial plantations of Norway spruce at lower heights, where it's not naturally occurring, and this has been done from 100 years ago to 50 years ago. So with the goal of producing very good timber for the economy. So then with climate change, these plantations proved not to be the best. So now there are big problems with bark beetle infestations, great damages in all Central Europe. And all the foresters are dealing with it.
First of all, great presentation, and you show a lot of action about to sustainable forestry and nature protection. And I'm curious what people think about this action. If they believe that Slovenia is doing enough for nature protection, or there are some discussion or conflicts with the public. Because you also do some policy action and education, so maybe you know what are the public awareness about this problem.
Yeah, so public perception is always a bit tricky to measure. But we had studies done, and studies by different researchers, and then the main national studies don't always show the same results. So I guess it's like most places, some people are happy with the forest management, some are not. But so far, the public support for this forest management system is still quite high. So I think the problem is more in the future. I did last year just a little poll with school children, and they really believe strongly more in conservation and not so much in sustainable forest management. But let's say the large studies that were done were like public barometer, okay, about public opinion, really support active forest management in Slovenia.
First, thank you for your nice presentation. As you mentioned that climate changed on forest, and I want to ask you, have you checked how the morphologically changed the forest, or in another word, how changed the intensivity of the fragmentation? And my another question is, have you checked the priority area? If yes, how much there is the overlap between this priority area and the already protected area?
Thank you. So morphologically, I hope you mean like stand structure, and climate change is making the forest younger, which is good, as we saw. But it's not the best that you do salvage logging and not proactive forest management.
How change the fragmentation? How have you checked how the climate changing the fragmentation in the future?
I wouldn't say there is much additional fragmentation because there is so much forest cover. Otherwise, it would be different.
So have you any plan to vast your protected area, or any some construction for the future?
I'm not sure. There are many protected areas. 46% of forest is in Natura 2000, and there are the ecological cells of the FSC. It's a network, and some also other local protections because of nature conservation. So we are not, let's say, well, it's again, this is not opinion. This is my opinion, but I don't think a lot of additional protections is necessary. But many would disagree, also in Slovenia.
Thanks. I think that the biggest problem is that we are not sure, and we will never be sure, because the trajectory of climate change intensity will differ, and it will be always projection, what will be next. So what would be the wisest solution? to split the risk. To split the risk not only across the species, because we have a lot of sources of uncertainties. I mentioned some of them in the lecture yesterday, but also data deficiency from some regions can affect it. And another source is we don't know which strategy of management or not management will be helpful or not helpful, because the question is what we want to achieve from forests. If you look to the recent papers from Loud Saint and from another person, from Nature and from Science, they showed the trade-off between forest carbon sequestration, albedo of the forests, and forest microclimate. And also with the economic forest value. So I think that the optimal strategy would be a mixture of forests aiming to store more carbon. Also, you need to account the fact that not only woody biomass stock carbon, especially in the wetland areas where the organic soil can stock much more carbon than the stand. And in such cases, it would be wise to not include any forestry interventions, just let it go, because it would be more stable. And also to include strategies to increase the carbon in the biomass, to conserve biodiversity, because sometimes there are also trade-offs between what we want. There's a lot of studies, especially from Scandinavia. In recent years, there's even a special issue of Ambio about the optimization, about the mathematical methods to split the particular forest parcels into different simulations of management options. And using these simulations, it is easier to select a country scale strategy that will be optimizing a particular forest compartment for particular function. And I think this number of Ambio would be a good point of view for your further studies.
Thank you, Marcin, for your comments. Guys, can you?
I'm not sure that I have a comment. It's just some very good advice. But it's always a question of segregation or, you know, doing what's the opposite of segregation? Integration, yes. Of course, when you segregate, a lot of things are easier to calculate. But I think a forest will always be a black box. So there is always going to be something that we are not going to be able to, that is going to surprise.
Once again, thanks. I think that we can move on to the questions for Marta Kujawska about humus and the mycorrhizal association of these three. Do you have any questions? Yes?
First one is some part of your first study I missed. So you didn't find any difference in different seasonal difference. You didn't find any seasonal difference, right? So, but in beginning of your study, you were expecting the seasonal difference or not? Yeah, beginning of your study.
Unfortunately, I know that the season is very important factors for fungi, especially for symbiotic fungi, but unfortunately in our research we can confirm that. Yeah, I know what do you think, because the season for you was very important factor.
Seasonal difference, everywhere seasonal difference is very strong because plants have difference in metabolic activity, different photosynthesis. So you can expect the seasonal difference. And my second question is that why didn't you use the AMF specific primer for glomeromycota? No. Why?
First of all, I don't have enough financial support to use primers for glomeromycota and ectomycorrhiza. So the main focus on our research was to find ectomycorrhizas on ulmus. Why? Because 10 years ago in our laboratory, my colleague found. So now when we have an NGS metagenomic analysis, we want to confirm that.
I can have some suggestion for for exploring the arbuscular mycorrhizal community. You can use the nested PCR method. I mean, 2 set of primer you can use. In the first step, you can design a arbuscular mycorrhizal specific primer and a universal fungal primer. Okay, and from the sample taken from the first PCR, you can use it for the second PCR. So in second PCR, you can use a arbuscular mycorrhizal primer and a universal eukaryotic primer. So this can give you very high abundance of AMF from my experience. Because in my PhD, I tried first with the ITS primer, but I found very low abundance of the AMF. Then I try with this nested method and find very high abundance of AMF, sequence abundance. Thank you.
So the second part of this research was we using the nested analysis and we tested the roots, not soils, and with the using the specific primers for arbuscular and for ectomycorrhiza. So we have 2 different words.
I would like to say that I liked very much the approach that you went to identify the problems with arbuscular and ectomycorrhiza, because you joined the field observation with the experiment to show that. And I have a question. What do you think about using this scheme to carefully and methodologically rigorously check the presence of ectomycorrhiza in Prunus serotina? Do you think that this would be a good option to test that? And if you think that yes, I would encourage you to test, because there is a debate.
So Prunus serotina is the object research of my colleague Robin Wilkan. So what I can say about Prunus serotina in the context of soy fungal community, we know that Prunus serotina have not negative impact on soy fungal community, but Pseudoacacia negatively huge impact. So. Yeah, and also we didn't have enough financial supporting, but what I say, that was based on soil samples. So maybe if we have money, we can check research what is inside the roots of Prunus or Pseudoacacia, and I think that will be very, very good next steps to research this word.
I wish for money to you.
I need
Marta, do you still trying to find the ectomycorrhizas using morphotyping? Because it looks like that I'm the troublemaker who found these ectomycorrhizas on roots of ulmus. And I think that was more than 10 years ago. And since that time, nobody found it. So do you use still this morphotyping to find one single mycorrhizas on ulmus oak roots?
Thank you for your support. Yeah, we really try. We started our research 3 years ago, because I have financial support from National Science, but unfortunately we didn't found. But we have something like a pseudomycorrhizas. You know, yes, but we need now more molecular analysis to confirm that. So maybe not ectomycorrhizas, but something between. Yeah, need more time and I don't know what.
Thank you for all questions about the humus and mycorrhizal fungi. Do you have any questions for Marta, but not Marta Kujawska, for Marta Górska about chemistry of soil, wood?
Could you remind me quickly how many samples you have?
From each of 20 trees on each plot, we collected 2 cores.
You showed a lot about statistical significance, but in such small number, I would rather focus on effect sizes and to show how much higher or lower was concentration, because this is the effect of this cemetery factory, and it would be more convincing, and I would rather show how much more was than focus on p-value, because due to small sample size, okay, you are not able to open another cemetery factory and wait 60 years to get similar effects, so you are using what you have. And in such a case, it would be easier to you to show more about these differences between means, and I encourage you to not focus too much on p-values. Thank you.
Thank you for your suggestion. Maybe I will add it to the statistical significance differences. Thank you.
Because of this pollution, do you found any other symptoms, like in the crowns, in the needles, or other specific situation in this forest, like less ground vegetation or something?
As I said, it was a part of the research. So yes, we investigated the increment course and the growth signal from the pines from Kunda, so from the polluted area. In general, was not so strongly affected as we supposed it to be, but it was not as unique as as homogeneous as in the control area. So the harm caused by the pollution was mainly visible in the different response of the singular trees in the polluted area. So they stopped to speak in a common voice, as I would say.
Thank you once again. Any more questions for Marta? If not, we may move to the first speaker. And Basil, do you have any questions for restoration of Pacific forest?
Though mine is not a question, but probably maybe a new research insight. So, because in your findings, you show that soil that are treated with organic manure show better result than compared to the untreated soil. So I want to know, like maybe if you have done it before, or probably it will be a new insight. Can we test for if for inorganic fertilizer and that of organic manure, if there will be any differences? So maybe to help us to bring more evidence as to better environmental option of organic manure.
Well, as I said, this data was not don't published yet, but my dissertation, my thesis, master thesis, I published the article, and in my mail research, I perform a soil analysis, and we see that the green manure changed the chemistry in the soil. So increase the magnesium, calcium, and other elements in the soil. And we also did another experiment to prove that this green manure can support the plants against the abiotic stress over the day. So at midway, when with much light and high temperatures, plants grow with this type of green manure is supposed to protect against the abiotic stress. So I did some analysis, but it's a short data from my master thesis. So thank you for.
Thank you for your presentation. I have one question regarding sustainability. In your soil treatments, you have a soil disturbance, which is against soil conservation. So, do you think this is ecologically viable with respect to sustainability?
We use these techniques to decompact the soil because we have a specific problem. This area have much traffic. We have machines, so the soil is extremely compacted and avoid the root the development. So our the main aim to the soil treatments is decompact the soil, and the green manure ensure that change the chemistry in the soil to ensure the seedlights will be grow in that environment without human intervention after.
Yeah, you've shown that your treatment is effective in improving plant condition. My question is how general is that method? I mean, how much land of this type you have for to reclimate in Brazil?
We performed this experiment in a tropical area using some species of beans and leguminous species. But I think these treatments and this type of green manure can change a little bit to use in other types of climate. But we need test before because we test for a specific solve a specific problem in a specific climate and soil conditions. So we must test before to use this type of treatments in other areas. But yeah, yeah.
Okay, I think that we may finished our session and słusznie? Okay. Now I invite you to the lunch break. Thank you once again.
Please come in. Okay, welcome everyone to this afternoon session. My name is Daniel Chmura. I will be hosting this session. We have 5 talks. One will be in a form of recorded presentation. I hope everyone knows the rules, so please keep in time so that to allow people switching between session rooms. I just noticed in my agenda that after our session there is planned coffee break, but after the session in the other room there is lunch. So if you didn't have enough, please switch rooms. Okay, jokes aside, the first speaker this session is Salvatore Francesco Papandrea with the talk entitled Noise Propagation in Forest Environment During Cross-Cutting Operations with Chainsaw. Yes, please.
Take the floor. Good afternoon, everyone. I am Salvatore Francesco Papandrea. I fellow research at the Department of Agraria of the Mediterranean of Reggio Calabria. And today I would like to present this research work entitled Noise Propagation in Forest Environments During Cross-Cutting Operation by Chainsaw. Many of us are already aware that the wood from forests is the raw material for the many everyday products. Wood for our homes and furniture, paper for our books and magazine, packaging for our wood, and wood bike, for example. The potential of the Italian forest heritage is notable, thanks to the favorable station condition, which allowed an extension of the vegetative period with consequent benefit in terms of the increase the level of the production forestry. The Calabria region occupies a 50th place among the region of the Italy within about 600,000 hectares of forestry area, and a woodiness index about 14% according to the data from the National Forestry Inventory. The sensitive area in Calabria region was subdivided in the 3 national parks, 1 regional park, state reserve with about 2,000 hectares, regional state over areas, regional reserve, and sensitive area inside to the network Natura 2000. With 6 seen and seer, about 35 hectares. Generally, the Calabria region occupies a strategic position in the center of the Mediterranean. In fact, there is the port of the Gioia, which is the main Mediterranean tranship port. And today it's among the important in the world. In my opinion, this element can enhance the market of the Calabria region, and in particular can enhance the wood market. Forests represent an environment delicate ecologic balance, where activities related to the wood harvesting increases like generative environment disturbance, such as soil compaction, air pollution, in the case of this study, noise pollution. Over last century, the forestry sector has seen progressive growth in the mechanization of operations starting after the Second World War. The intervention primarily concerning the felling and the cross-cutting of the trees, for which the motorized chainsaws was introduced to replace axe and saw. In the past, human presence in the forest during wood harvesting or forestry operation causes slightly sound, such as footsteps, creaks, verbal communication, and the axe blow. However, with the advent of technology, and in particular with the introduction of the engine, the noise is more noticeable. Forestry operation often involve the use of the equipment and the machinery that can produce noise and pose potential, permanent or temporary disturbance for forestry worker and wildlife. High noise level, in fact, produced by the use of the census in the forest can interrupt the natural cycle of animals, such as eating habits, mating and migratory paths. Exposure to noise can cause the extinction of some species of animal in a noise-polluted environment. For this reason, the work aimed to carry out a first experimental approach to assess the range intensity of the noise generated by census with different powers, distinguished in terms of the lower power census, medium, and high power census. By applying an appropriately set up measurement system in order to create a realistic condition. The study was conducted in sensitive forest area of the Sila National Park in southern Italy, where a pilot simulation of cross-cutting was tested on European beech and the Corsican pine logs. In particular, this research aimed to evaluate the human impact on environmental sensitive area in order to gain insight into the phenomena of the acoustic disturbance and to mitigate the negative ecological impact of forestry operation. The simulation consisted of wood cutting discs of the same thickness from logs, with the logs with the same diameter, and using different census but the same census operator. The assessment of the noise propagation during cross-cutting was carried out with 16 sound data logger, which recorded sound level in a geometric pattern planned in the test year. Each data logger was mounted at 2 meters above the ground and positioned at 4, 8, 16, and 32 meters away from the operator. The area was subdivided into 4 cardinal points, north, south, west, and east, and the measurements were carried out in a coniferous forest stand, such a way to recreate realistic condition of forestry site. The research protocol involved in different steps. 3 different census distinguished by engine power, maximum engine during cross-cutting for each grip, 2 different log property, hardwood with European beech and softwood with Corsican pine, and 16 sound data logger class 2 to record the acoustic level located in a geometric pattern. From the preliminary result obtained regarding the softwood, the graph showed that the lower power census during cross-cutting of Corsican pine logs produced the highest noise level. And for hardwood, also in this case, and more clearly, from the graphic can be seen that the lower power census in European beech logs produced the highest noise level. By comparing the noise value found during cross-cutting of the log with different census, it's possible to see how the wood species influence the amount of the noise generated by the census. It would be expected that cutting article would result in higher noise level, but this result was only observed in the case of the lower power census. In the contrast, softwood generated by the slightly higher decibel when cutting medium and high power census. Analyzing more specifically the propagation of the noise in the forest environment in which the experimental cuts were carried out, a notable reduction in the sound reaching the microphone positioned at 32 meters away from the source was observed. For both types of the wood, it's possible to observe how the noise of the lower power chainsaw decreases less than the other two types of the chainsaws, even at a distance 32 meters from the source. This trend could be due at an increasing time in the cutting of the individual wooden disc of the lower power chainsaws, which probably caused assumed the individual sound wave and therefore a greater frequency and intensity of the sound, even to a greater distance from the source. Comparing chainsaws of the same power, we can see in forest conditions the noise level is reduced by the same percent as the distance increases, regardless of the species cut. The trees in the forest served an acoustic barrier to the sound wave generated by the chainsaws. Among the similar results, it can be observed that the lower power chainsaws have a slightly lower reduction as the distance increases compared to the other type of the chainsaw. In conclusion, this study confirmed that the chainsaw is one of the most critical machines in terms of the noise level in natural environment. And in this preliminary study, it was observed that the intensity of the noise significantly decreased moving away from the source. No significant difference were observed between hardwood and softwood, with the exception of the lower power chainsaws, which probably due to the long timer to need to cut the wood disc, especially for hardwood, caused a higher sound pressure and therefore no intensity even to 32 meters away from the noise source. Some studies have shown that some animals, such as spotted owl, exhibit psychological change when exposed to sound pressure level about 16 decibels, just for 10 minutes, generated by the chainsaws. Considering the simulation era and the study carried out on similar era, the modest influence of the tree density and the noise attenuation, it can be deduced that the operator working around the chainsaw operator, can consider himself in the safe position if his distance exceeds to 12 meters from the noise source. The study showed the potential of the predicted noise exposure generated by cross-cutting operation by taking into account a specific ambient condition. The phonometric data processing has enabled to outline the typical representation, even to approximate. The results could be used to illustrate the possible extent of the area affected by the noise from multiple sources in the forestry site, and the advantage of the predicted noise propagation in forest operation can summarize it in supporting the identification of the most environmentally friendly work system in terms of the noise propagation by taking into account technical and logistical contrast, for example, terrain slope, skidding and road network, forestry site management. And identifying the area where exceed noise level for the different frequency band can negatively influence the ecological response of wildlife species. Obviously, it's possible if the data are variable on wildlife response to noise. Thank you for your attention.
Thank you very much. Our next speaker is Jolanta Masłowiecka with a presentation, Study of the Chemistry of Leaf Litter Decomposition of Selected Deciduous Trees in Environmental Conditions. The presentation is recorded, so we'll just watch it.
Good morning. I would like to present some of my results of my doctoral thesis titled Study of the Chemistry of Leaf Litter Decomposition of Selected Deciduous Trees in Environmental Conditions. In the first dictionary, litters is undercomposed plants remains of the forest floor. It is assessment that lighter fall in coniferous forest is 7 tons per year, and in deciduous forest 4.5 tons per year. On the global scale, this gives us almost 194 billion tons. Ritual decomposition contains cell mutation processes, mineralization and humidifications, leaching of soluble compounds into the soil, release of volatile organic compounds into the atmosphere. These processes depend on abiotic and biotic factors. These processes have been studied to varying degrees today, with the third being the last studied. Therefore, in my work, I pay special attention to examining the composition of dynamics of VOC emissions from disposing garbage. VOC are unusual and have attracted scientific attention since the 1950s, when it was discovered and that the oxidant in the atmosphere lead to the formation of photochemical smoke. Most VOC emissions are highly reactive and affect atmospheric chemistry to a greater extent than organic substances from industrial and motor vehicle emissions. Further research has shown that VOCs are involved in the formation of substances with toxic and phytotoxic effects, such as ozone and peroxides, and they participate in the formation of secondary aerosols and influence the radiation flux in the atmosphere, and consequently influence the radiation budget and troposphere. The graph shows that the dynamics of ozone concentration grow. Measurements were carried out in European mountain stations in the 20th century and beginning of the 21st century. Ozone is a greenhouse gas, and its concentrations in constantly increasing. It has a significant impact on human health as well on vegetation and climate. It is well researched. A variety of particles suspended in the atmosphere constitute secondary organic aerosols. They also affect atmospheric chemistry and human health. They can scatter and absorb solar radiation and also act as cloud condensational nuclei. For example, a photo taken in the Blue Mountains National Park located near Sydney, Australia. The name of the park comes from the eucalyptus trees growing there. The essential oil contained in the leaves float in the air as a result of evaporation, creating a slightly blushing mist. The inventory of anthropogenic nitrogen oxide emissions has now been complete. The situation is worse in the case of VOC emissions. The main source of VOCs is considered to be the living vegetation of continents. Plants emit them in the process of respiration. Plants emit VOCs for a variety of purposes. They are defense compounds against pathogens and herbivores, and signaling compounds between plant and other plant parts. They are emitted in response to injury and stress. When garbage decomposes, the production of VOCs is 10 times greater than the production of abiotic VOCs, and pathogenic emissions are estimated at 450 million tons per year. Changes in the composition of the global atmosphere that have occurred in recent years have shown that existence of the so-called lost sources of VOC. According to many experts, the missing source is the emission resulting from the decomposition of garbage. Much attention has been devoted to the study of this process, but they mainly concerned coniferous forest litter. When it comes to the litter of the deciduous trees, the literature is extremely poor. In my work, I decided to explain and partially fill the gap in our knowledge about the process taking place under the forest canopy. During the experimental solution to this problem, it turned out that the litter contains large amounts of soluble organic compounds, but there is no information in the literature about penetration into the soil as a result of leaching the partially decomposed leaves. This was used to formulate working hypotheses and main research objectives. Hypothesis. First, the release of organic compounds as a result of litter decomposition is bidirectional and affects both the atmosphere and the process occurring the forest soil. Second, decomposition of VOCs and compounds leached from garbage is a mixture of components of various origins. Third, emissions from pollen inflorescences of woody plants are a previously unknown source of high reactive VOCs in spring and summer. Goals adopted at work. First, determination of the chemical decomposition of VOCs emitted during decomposition of Birch, Alder, hornbeam, oak, and aspen leaf litter. Second, study of changes in the chemical decomposition of volatile and extractable organic compounds during the annual cycle of leaf litter decomposition. Third, determination of the chemical composition of volatile and extractable organic compounds in pollen inflorescences of forest-forming tree species. The research material used came from dead leaves of oak, birch, aspen, alder, and hornbeam. In autumn, yellow leaves were collected directly from trees or those that had fallen in the city of Białystok or its surroundings. In the laboratory, the leaves were dried in the dryer at 50 degrees until constant weight was obtained. Some of the leaves were transported to the Koszyńska forest to the Arboretum in Kopna Góra, where 4 research plots were established. One plot for black alder was established in the Wierzchniecki Forest Nature Reserve. Due to the complex nature of the tested material, various analysis techniques were used. To examine the rate of precipitation distribution, their mass losses were examined using the cut-circle method. The chemical composition of volatile compounds was examined using the solid phase extraction method, and the chemical composition of non-soluble substance was examined using a GCMS apparatus using 3-stage solvent extraction. Hexane was used to obtain non-polar substances. Then ether was used to obtain weakly pure substances. And finally, methanol was used to obtain polar substances. To investigate the organic compounds leached by rainwater, the SPE solid phase extraction technique was used. The chemical and biochemical oxygen cement permanent was used as a quantitative injector of dissolved organic carbon content. The study also examined inflorescence litter. When examining soluble compounds, the mentioned technique of soil-based microextraction and methanol extraction with the polar solvent were used. VOC emission was examined for fresh litter and after 6 months of exposure. The largest group were terpenes, monoterpenes and sesquiterpenes, both in fresh leaves and those exposed for half a year. The second group included alkanes and alkenes. There was an increase in the content of terpene compounds in the dead Birch litter, which was most likely caused by prolonged hydrolysis of glycosides, because most terpene compounds in plants occur in the form of glycosides. An increase of content of carboxylic acid was recorded in the stall litter. Methanol was detected, and according to reports by other authors, it is a product of decomposition of cell walls by microorganisms and physical environmental factors. In carbonyl compounds and aliphatic alcohols, compounds typical for many species of mushrooms have been detected: keton 3-octane and 1-octane triol, named mushroom alcohol. As a result of various processes, significant amounts of aromatic compounds appeared in the old litter. The more aromatization of VOCs occur as a result of hydrogenation of terpenes or the appearance of oxidized compounds. The content of aliphatic acid decreased, perhaps due to the depletion of sugar readily available for microbiological absorption, which act as the main acid producers. The old litter contains sulfur and nitrogen compounds in very small amounts. These compounds are most likely of microbiological origin, as the production of these substances is characteristic of many microorganisms. Shows the average COD values in water washed from fresh litter during 3 rainfall episodes. COD values varied depending on the type of litter. The lowest values were recorded for Birch litter, the highest for black alder, and in 2 cases for aspen. This difference may result from the intensity and duration of rainfall. In all cases, it can be concluded that the flow of rainwater through the litter resulted noticeable enrichment in organic carbon. The difference between unfiltered and filtered water, for 14% of average, shows that some of the organic carbon washed out and fermenting through the litter consists of composition of leachate particles. Compounds were divided into 7 groups. The content of individual groups was not very specific, with the exception of Birch litter, in which a higher content of terpenoids was recorded, while in the remaining litters there were none at all. A high sugar content was recorded in the tested litters, which indicates the environment of fungi, bacteria, and invertebrates that participate in the composition of organic matter and produce their own specific substances with varying degrees of volatility. Some of the results were published in a recent publication in the journal Geoderma.
I guess that's it. Okay, thank you so much for watching and listening this presentation. Okay, let's move to the next speaker, who is Ricardo Di Cintio, with the talk Optimizing the Assessment of Habitat Trees in Forest Landscape Management Planning.
Thank you. Hello to everybody. I am Riccardo Di Cintio, PhD student at University of Tuscia, Viterbo, Italy, and it is a pleasure to talk to you about habitat trees in the context of forest landscape planning. Translating sustainable forest management and especially environmental policies related into action on the ground requires an adaptive landscape approach that considers fully different land ownership structures, interests, and values involved. In turn, forest landscape planning requires strong decision support tools, such as those offered by multi-criteria decision analysis. However, it is questionable that forest landscape management planning can address the influence of management on the diverse forest functions without a stand-level indicator that work as interface between forest model and the decision support system. Regarding biodiversity conservation function, habitat trees combined with other indicators enable or could enable the evaluation of forest management effects. In literature, we can find several methods to assess habitat trees, but in general, they are very complex methods suitable for the research with a wide array of TREMS type to related microhabitats, requiring additional efforts and cost for data collection. Moreover, they are characterized by unclear management outcomes, and here we can see some example from ASBEC based on the classification of Larrieux and the method proposed by Krauss. So in order to overcome these practical issues of the existing methods, we propose a novel habitat tree assessment procedure called radar table, where multiple types of tree-related microhabitats are aggregated into wider categories. And the table considers 5 types of tree-related microhabitats, plus a risk assessment evaluation of the tree, and the aspect of characteristic in order to take into account some special feature like pollarding, bifurcation, rarity of the tree species, and the cover rule. The ecological value of each for each of those aspects is assigned a score, and the ecological value of each individual tree is determined by the sum of score obtained and classified as shown in the table 1. Then it is calculated a virtual population per hectare of each tree thanks to the Relascope technique. And the sum of the products between the number of habitat trees per hectare as classified according to the radar table and the score defines the ecological value of the stand per hectare. So we apply this method in this study area, that is the natural reserve of Vico Lake, located in Viterbo, Italy, where we conducted a systematic sampling in European beach at Turkey Oak ice stand, using the radar table and collecting forest and topographic measures. We analyzed tree-related microhabitats richness, abundance, and density, and performing generalized linear models with negative binomial distribution instead of the suggested from other study Poisson distribution, because we observe problem with over dispersion that affected the Poisson distribution. And ANOVA was employed to assess the ecological value of the stand efficacy in stands discretization. And in addition, we verify its independence from common forest metrics. For the plot factors, we discover significant association between quadratic mean diameter, canopy cover, forest stand composition, and the forest district that serve as a proxy for different management systems. Stand density exhibit a negative correlation with abundance, whereas density show a positive correlation with slope. In terms of tree factors, we noted the significant relationship between tree-related microhabitats abundance and richness with DBH and tree species. Specifically, we observe a decrease in abundance starting from other broadleaf that comprises Acheropalus, Ostea carpinifolia, Carpinobetulus, and Fraxinus ornus, and to European beech and finally Turkey oak. Okay, the ecological value of the stand. Regarding the ANOVA reveals a significant association with the forest district and so with management systems, while forest and topographic covariates show minimal variability. Furthermore, no correlation is observed between the ecological value of the stand and standard forest metrics. These results are only just for the European beech stands. Here we can see on the map the spatialization of the ecological value of the stand of the study area, and so consequently it is kind of the representation of the density of habitat trees in the study area, where we can observe a much higher value for the European beech in confront of the respect to the Turkey oak stands that derived from the conversion of copies. And here in the graph, we can see the distribution of the ecological value on the stand among the DBH classes of the habitat trees. So what we can say, the DBH and tree species are main drivers for tree-related microhabitats, abundance and richness, but they are not enough to identify an habitat tree, as confirmed by other studies. So what we can take here is that the abundance and richness of tree-related microhabitats seem to be species-specific. And regarding the plot, the influence of plot factors, we have significant association, as we said about before, with quadratic mean diameter, stand density, canopy cover, and composition, and the forest district. While the topographic measure, just the slope, shows a relationship with density of tree-related microhabitats. Regarding the second point, I think that the most important thing is that the abundance and richness of microhabitats depending most on management system, highlighting that the lack of trees, habitat trees in managed forest largely stems from silviculture practice that have not yet considered these aspects. And about the radar table, we think that allows to assess efficiently, based on this case study, the habitat trees density and consequently the ecological value of the stand of a specific forest district that seems independent from standard forest measures. And the preliminary result affirmed the radar table method's validity in optimizing survey procedure, and this could facilitate the operational forest biodiversity conservation and its integration into forest landscape management planning. And plus, I would add that also I think that is the kind of effort that we have to do in order to harmonize the forest inventories across the European Union that are start to take into account this kind of tree. Thank you.
Thank you very much for your presentation and an excellent time regime. So we are even ahead of time. But let's move on to the next talk. Please welcome Natalia Mazurek with talk The Influence of Environmental Variables on the Species Composition and Richness of Oak Forest Stands in Large-Scale Scots Pine Monocultures. Please.
Hello everyone, my name is Natalia Mazurek. I'm a PhD student at Botanical Garden at the University of Wrocław. Today I'm going to talk about the influence of environmental variables on species composition and richness of oak forest stands in large-scale Scot pine monocultures. Forests occupy 30% of the area of Poland and Europe. Most of them are managed for commercial purposes. The species composition of tree stands in lowland is dominated by Scot pine. In contrast, deciduous forests, which are considered to be natural vegetation of European temperature zone, occupy a significantly smaller share. In these forests, pedunculate and sessile oaks are the most important species from economic and environmental perspective. They create tree stands in wide variety of forest types, from fertile oak hornbeam and riparian forests to poorer acidophilous oak woods and oak pine forests. Aim of the study was to assess richness and composition of middle-aged and old oak forest stands, as well as to identify environmental variables that influence of vascular plant diversity. The field research was conducted in Borys-Tobrawskie mesoregion in southwestern Poland. This area is representative for lowland forests in the temperature zone. Moreover, forests in this region consist of large complexes and cover over 50% of the land area. Field research was conducted between May and August. Data were collected from 100 circular research plots divided into stand age groups. Middle age stands 31 to 80 years and old stands 100 to 108 years, with an amount of native oak species over 15%. On each research plots, a list of vascular plants species occurring in all forest layers with an estimation of the cover was made. Data from research plots were divided into groups according to stand age, site productivity, and site moisture. During analysis, species richness was counted for each plot and stand group. The species composition of these groups was compared using ordination methods, and we also made a list of indicator species, taking into account the fidelity coefficient. Finally, we assessed the influence of environmental variables on species richness, composition, abundance of forest species groups, and species habitat preferences. In oak forest stands, we found 257 vascular plant species during the study, 208 in middle-aged stands, and a little more, 215 in old stands. The mean species richness was slightly higher in the middle-aged stands in the whole data set and in the analyzed productivity and moisture groups. But the highest richness was found in the old forest stand. It was 74 species per plot. Differences in the average species richness of studied groups were not significant, but it can be noted that regardless of age, of stand, more species are occurred in fertile and moist habitats, and in poorer and fresh sites there was a lower number of species. This is not surprising because fertile and moist soils favor the occurrence of vascular plants. As the research was carried out in forest areas, we paid a particular attention to the proportion of forest species in the total species pool. In all oak stands groups, the species that can occur in both forest and non-forest sites were the most common, and species associated with close canopy forests were the second most common. But there is no significant difference between mean participation of forest species in oak stand groups. However, there was a trend in the total number of species, of forest species. In old stands, so old stands had more species associated with close canopy forests and fewer species associated with forest and non-forest habitat than middle-aged stands. The exception was stands in poor habitats. Influence of environmental variables. Species richness decrease as the age of oak with the age of oak stand. This may be related to the gradual disappearance of light-demanding species, which are more abundant in younger and disturbed stands with incomplete canopy closure. Species richness also decreases as the proportion of oak in stand increases. This phenomenon can be explained in two ways. Firstly, a higher proportion of oak means fewer other species in stand, and this can have a direct impact on species richness. Secondly, more monotypic stands are characteristic of poorer systems, such as acidophilus oak woodlands, and in contrast, more varied stands are found in more fertile forests such as oak hornbeam forests and riparian forests. Additionally, species richness decreases as the proportion of beech in stand increases. Beech is a tree known to be a heavy shading species, and their fallen leaves take a relatively long time to decompose, which can potentially limit the development of underground vegetation. In addition to stand characteristic, the location of vegetation patch also affects species diversity. There is a general decrease in species richness moving northwards. I hope it can be seen on the map. So species richness was marked with dots from white to dark red. On the north, the species richness was lower than on south. Moreover, large patches of all deciduous forests contain fewer species on average than smaller patches distributed among Scott pine monocultures. Both stand characteristic and location have also influence of the proportion of species in forest species groups. Nevertheless, the sheer number of species is not the only criterion to be considered. The composition of species is also important. The influence of environmental factors on species composition was evaluated using the ordination methods. Presented diagrams demonstrate that habitat moisture has the greatest influence on the variation of species composition. However, stand age and site productivity also has important impact. Middle-aged stands dark dots differ from old stands with the species composition. And when we look only on poor sites, dark dots, and fertile sites without intermediate sites, we can see clear differences between these two groups without these intermediate sites. The most significant factors influencing species composition are oak age, latitude and mean annual temperature, annual precipitation, altitude, and area and shape of oak of deciduous stands exert a larger influence. Due to the occurrence of differences in species composition, a list of indicator species was created of each oak stand groups based on the fidelity coefficient. It can be observed that old stands on fertile habitats have much more indicator species than middle-aged stands on fertile habitats. And opposite, on poorer sites, more indicator species were in middle-aged stands. Moreover, groups of similar fertility and age have some individual species in common. They are marked with the same color. Furthermore, a comparable list was compiled for groups differing in age and site moisture. In both age groups, moist sites have much more indicator species than fresh sites. Additionally, old stands exhibit species more specific to habitat type, while middle-aged stands exhibit species with more average habitat requirements. The occurrence of species with different habitat preferences in the list of indicator species provided the rationale for comparing groups of oak stands in terms of mean values of ecological indicators. The Kruskal-Wallis test and post-hoc test showed that habitat preferences of species found in oak stands on habitat with different productivity were different. Furthermore, differences in species habitat preferences was also observed between stands on different moisture sites. However, there was no significant difference in species habitat preferences between old and middle-aged stands. The habitat preferences of species have been found to vary according to stand structural characteristic and the location of vegetation patch, as evidenced by statistical tests. To sum up, I can say that all stands are favored by species of dense forests. Patches of old stands are more variable than middle-aged stands in terms of species number and the proportion of species groups. Species richness and composition depends on stand characteristic and location of the vegetation patch. But other variables have the greatest influence of species richness and composition. For richness, there are stand age, proportion of oak and beech and stand, latitude and size of old growth patch. And for species composition, it's age of oak, latitude, and mean annual temperature. It is evident that habitat conditions exert a greater influence of species composition than stand age group. Further research is required because the species richness of vascular plant in old and middle-aged stands exhibit only slight differences, the majority of which were not detectable by statistical tests. We have already started research that concerns on diversity of bryophytes and lichens occurring on all available substrates in the studied oak stands. Thank you for attention.
Thank you very much for your presentation. And our last speaker in this session is Sonia Paź-Dyderska with a speech, How much do the floral, fruit and leaf traits of woody plants vary within a species?
Okay, so hello everyone. My name is Sonia, and today I would like to tell you about the variability that occurs in floral, fruit, leaf, and stem traits of plants within the same species. The idea for this research occurred from a major concern, that is, under changing climate, we need improved methods to predict how the future environment will look like. And one approach to improve such predictions is using plant functional traits. But what are plant functional traits? These can be morphological, phenological, and physiological characteristics of plants that describe their growth, reproduction, and survival. Some examples include, for example, height or seed mass. And also, the big benefit that comes from the use of plant functional traits is that they are also intercorrelated. That means that knowing the value of one trait, we can sometimes predict, assess the value of other traits, perhaps ones that are more costly or more time-consuming to measure. And these traits can be utilized for quantitative comparisons of life strategies among species or for predicting changes that will occur in species composition or plant communities. However, there are also some drawbacks that comes when using plant functional traits. Firstly, the phylogenetic and geographic representation of species in databases is uneven. So what I mean here is that some groups are better represented. For example, trees or some species that are economically important to people are better recognized than, for example, herbaceous species. And geographically, there are more measurements for the global north compared to the global south. Also, there is a scarcity of repeated measurements of traits across many species, which lead to underestimation of intraspecific variability of traits. So even when we have measurements for a given species and a given trait, it sometimes occurs that it's only for one plant or several plants. And taking into account that the plants often have wide ranges of occurrence, then maybe we are not aware of their intraspecific variability. Also, most studies focus on the most commonly measured and used traits, as is it well recognized, and also the data is easily available. And finally, the recognition of traits across different plant organs is uneven. One of the examples of the less recognized traits are floral and fruit traits. Although they are crucial for reproduction, they are often sidelined in research. And the sole exception here is seed mass, which particularly to the Q database is very well recognized. And the conservatism of these traits could potentially mitigate methodological imperfections linked to utilizing average trait values from publicly accessible databases. So taking into consideration that they are less variable than, for example, leaf traits, maybe we could extrapolate measurements for a lower number of specimens to the rest of the specimens of a given species. So in this light, reproductive traits may harbor great potential in functional ecology, but we need to quantitatively assess their usefulness. So the aims of our study was to compare intraspecific variability of traits among different organs and to determine the organs with the highest and lowest variability within species. And secondly, to compare the intraspecific variability of traits across various plant organs with their interspecific level variability, aiming to evaluate the potential of floral and fruit traits for further studies. And we hypothesized that reproductive traits, so floral and fruit traits, would have less intraspecific variability compared to performance-related traits, like, for example, specific leaf area. And let's go to the methods. So we collected flowers, fruits, leaves, and stems from 20 species. And each species was represented by 6 populations. Each population, 6 specimens. By sampling individuals from 6 separate populations of the same species, we were able to assess the variability of the studied traits at the intraspecific level within species. And then we could assess interspecific variability comparing the values for each of the 20 species. So here you can see the 20 species that we collected, and then for each one of them we collected the 4 organs. It has flowers, leaves, fruits, and stems. For each one of the species, these 4 organs were collected from 6 collection sites, each one at least 5 kilometers apart from another one. And when it comes to the study process, from the collection sampling then to the lab, we have measured the organs, weighed them, scanned the leaves, grinded the material, and finally analyzed the content of carbon and nitrogen in them. Here you can see the map of collection site. In total, we had 118 collection sites. And here I wanted to show you a short video of me in the field, so you know that if you collect flowers and fruits, it doesn't mean that you don't have the opportunity to also get it into some quite dense bushes. And also there were some different factors that make it quite difficult to collect a totally full database. And here I mean insects, fungi, and also birds, because as one of my assumptions was to collect fully developed organs, sometimes what occurred was that when I went once to the collection site, the fruits were not yet fully ripened, and the next week they were already gone due to, for example, the activity of birds. And regarding the analysis, we have used the newly proposed coefficient of variation kind of analysis, which is useful for my kind of research because due to this formula, we get values from 0 to 1 and not some abstract numbers. So it's easy to compare the values of coefficient of variation for numerous traits. So let's go to the results. And firstly, we aim to compare intraspecific variability of traits among each other and with their interspecific level of variability. So here I wanted to show you an example of this kind of analysis for just one trait to begin with. Here we can see the level of coefficient of variation for specific leaf area at the intraspecific level represented by the green bars, and the solid line represents a mean value of coefficient of variation for SLA for all the species. And then we showed it for all traits using box plots and points representing each species intraspecific trait variability, and color bars representing each trait's interspecific variability of particular trait. So you can see that, for example, for fruit length and flower dry mass, the intraspecific variability is similar, for example, as in the case of leaf traits, but their interspecific variability is very high. So it's generally beneficial for ecological studies because we have species that produce very different species between species, but they are quite conservative within species. And we use GLMMs, assuming beta distribution of dependent variable, to compare traits, including the phylogenetic correlation between the species. And this model served us to compare mean coefficient of variation for each trait. And we found that the highest intraspecific variability was for fruit dry mass, then specific leaf area, and stem carbon to nitrogen ratio, while the most conservative ones were the carbon-related traits. Here you can see the phylogenetic tree showing that generally we attempted to include quite diverse array of species. And finally, to conclude about traits applicability, we plotted estimated mean values of coefficient of variation for intraspecific variability on the interspecific variability of each trait. So what we can see here again is that for fruit and flower traits, generally we have quite high level of interspecific variability, while the intraspecific variability is at a regular level. So let's go to the conclusions. And the traits with the highest intraspecific variability were fruit dry mass, specific leaf area, and stem carbon to nitrogen ratio, whereas carbon-related traits had the lowest intraspecific variability. Despite the significant intraspecific variability observed in floral and fruit traits, the ratio between their intraspecific and interspecific variability was significant, beneficial, confirming our hypothesis. And integrating floral and fruit traits into ecological studies can significantly deepen our understanding of plant life strategies and enhance predictive capacity in environmental science. This study was financed by the National Science Center as a part of a project Preludium and also supported by the Institute of Dendrology of the Polish Academy of Sciences. And I would like to thank to Kinga Nowak, Roma Żytkowiak, Marcin Dederski, and engineering staff from our Department of Ecology. Okay, thank you for your attention.
Thank you very much for a very interesting talk, and thank to all our speakers at this session. So I open discussion now, starting with the first talk from Salvatore. Are there any questions to that presentation?
As I remember, you measured the noise propagation. So my question is if you considered to include also as a factor the air humidity, if it can affect the noise propagation.
Okay. This is preliminary study. We measurement the humidity of the wood, the moisture of the wood, but not the humidity of the ambient. And we need to collect more data to expand the range of the noise wave propagation. And but this result is important because we not expected the lower power chances avec highest noise level. And this is important because in the deliverable in the future we use the electrical sensors. Now electrical sensors not have a FNC of the engine sensors, but when we cut the small diameter and felling the small tree, we use the electrical sensors, and the electrical sensors don't have a noise impact in the environment.
Any other questions? Maybe I'll move to the microphone.
Smart.
Okay, so any other questions to the talk by Salvatore? Yeah, this was actually the question I was going to ask, because there are a lot of factors that might affect noise propagation, like stand structure, for example. You did your study in the uniform conditions, so yeah, your stand was uniform, but there may be other stand structures that may affect noise propagation differently. Yeah. So.
Yes, we recreate the realistic condition with the scan pattern we make, but in the other condition we don't know how the noise propagation. But the trees is served an acoustic barrier,
and it's possible to collect the frequency of the noise because the wildlife response in different species of different frequencies the noise value. So I don't know.
Okay. Any other questions to that topic? Thank you so much. Okay. We can't really ask any questions about volatile organic compounds, unfortunately. Although it's a very important topic under changing climate. Okay, so the next talk was by Ricardo. Any questions to that?
Yes, I have a question, Ricardo. The radar method seems for me very nice, useful. Did you make a comparison? How much faster is make an analysis of microhabitats using the radar in contrast to the standard procedure?
Thank you for the question, but unfortunately we didn't measure the difference. I only can tell you that the standard procedure in Italy, that is a kind of assessment of habitat trees because it's much complicated for that, requires a lot of time because the number of tree-related microhabitats are based on the Larrier classification and they have a huge amount. So I cannot say to you precise data, but during the master degree thesis, I also used the Larrier techniques and for small amount of trees because the threshold of the diameter is much higher, also for the assessment, and the time is much more.
Another question, what about transferability of this method to other countries? If I would like to use your weights to assess the richness and ecological value of trees in Poznań oak forest, what would be a problem and how transferable is that method to other ecosystems?
I think that there isn't any obstacle because the method works fine with in ice stand, and Poland, as I can see, mostly has ice stand. Some adaptation is needed for the copies. I used this method for a professional work before during the PhD thesis, and that needed an improvement, an adaptation specific for copies. But in Poland, I don't think you will find any problem.
Thank you.
All right.
Maybe I.
Go ahead.
No, it's not a question. It's let's say a comment that for me even gets stronger your results regarding that the most important aspect for let's say veteran trees is management, because I think that some of the variables that you studied, I don't want to go inside the statistics. For me, it's okay. I don't want to discuss this. But many of these parameters for me were somehow correlated among them. For example, you told that the slope was a positive proxy of the more slope, the more habitat trees, if I remember correctly.
Mm-hmm.
And this is, for me, it's a sort of, is related to management, because probably in higher slopes, they stop the active management before, let's say, before, and so there are more habitat trees. So I think that really only management, also the species, for example, the species it's simple, it's similar. Probably, if I remember correctly, Birch had more habitat trees than oaks, if I remember correctly. The opposite. Okay. So also this factor is related for me to when 50, 60 years ago, they all converted the copies to high forest or they stopped to cut as copies. But it's just, let's say, pro your result is.
I understand that, and about the slope, I have to look into it more because besides what you said, that it's correct, in my opinion, it's one of the hypotheses because is correlated with slope, but also because we could observe a much accumulation of certain type of tree-related microhabitats, such as in the slope and in that area that you know it, there are pyroclastic rocks, and the faults could move to the formation of basal cavities, for example, more than in other aspects of that area. And also because, but this is just a field observation, in general, in steep slope, we observe, let's say, singular shape of trees, and that seems to improve the formation of quindi, so to say that maybe it's not just the management, but of course there could be a collinearity between the two terms. So that model that you see need to be reduced probably, and take out this a consideration.
Yeah. Okay, thank you. That was my notes too. Okay, and see it. So the next talk was by Natalia about environmental variables. Yes, please.
So first of all, thank you for the really nice presentation. And I marked that the latitude was the significant variable. Can you remember what was the range of the latitude on the area you researched, so from the southernmost to the northernmost?
Sorry, no, so I can't say it. But it was the whole mesoregion Borystobrawskie. So you can, yes.
Okay, there's more questions.
I will go more about this latitude, because the range of latitude was very, very small. And also the range of mean annual temperature precipitation was very small. I would rather not use it in that analysis, because it is a very small range, very small effect. I would rather look on the microclimatic differentiation, and this significance of the latitude could be an effect of the southern you have higher elevations, higher differences between elevations, the masking of the factors. And what I should start my my talking, I'm very, very happy to see the good piece of geobotanical work presented, because there's not much more people who are doing good vegetation surveys. So I'd like to say that I'm very happy to see that. And I think that if you, instead of using raw Spearman's raw correlations, but go into the models and GLMs with Poisson distribution for species richness, I think that these differences that you showed in the data, but not showed by the Kruskal-Wallis test, you would have. And I encourage you to do that to have even better results.
Yes, we have planned to use models, but I can't do it, not yet.
Feel invited. Okay. Any more questions to that topic? No, I can't see. So we can move to the questions related to the last talk about floral, fruit, and leaf traits and their variability.
Don't be shy.
Yeah, don't be shy. We can.
So, sorry if I didn't notice it from your presentation, but what was the key to select the 20 species you chose for the research?
Okay, so the key was very practical. Generally, we try to select as different species in the terms of phylogenetics as possible, but also, to be honest, firstly we had 30 species, but for some of them it was really difficult to find those 6 populations formed by 6 specimens producing both flowers and fruits and growing quite near to Kórnik and Poznań, so I could just travel there. And yeah, so generally, as these are one of the first studies of this kind, generally I just wanted to collect species that I was quite sure that they will be flowers and fruits. And also I used iNaturalist, a citizen science platform, just to check out whether these populations have quite a high probability rate of producing flowers and fruits. Yeah.
Yep. Any more questions? Then maybe me. Okay, so well, it's a great idea to study fruit and flower traits because they are understudied. I was just wondering, did you account for within individual variability? Because, for example, for leaf traits like specific leaf area, it may vary greatly within individual tree, for example, depending on the position and light conditions within the crown. And how about those other floral or fruit traits? I would guess they don't vary so much, but did you think about that?
Yes, so to be honest, this was the previous part of the project, which I carried out in the Kórnik Arboretum, because here I wanted to include as many species as possible. So I have a simpler, let's say, study design. I have only 1 specimen per species, but I have 79 species instead of only 20 as in this case. And there I have collected 2 samples per each specimen from the shaded and from the sunny parts of the crown. And yes, I have also discovered significant intraspecimen variability across these traits. This was in an analogous way as in the case of this field work experiment study, let's say. So yeah, I think this will be nice and complementary once it gets published.
Yeah, we're looking forward. Good. Please go ahead.
Just a short question: how distant were these plots to each other for like one species? Like, yeah, how many kilometers or?
At least 5 kilometers apart. Yeah. Please.
Maybe just one more. So, initially, I wanted to make it 10 to 15 maybe, but then one factor that I didn't mention that make it quite difficult, especially in terms of flowers, was the crazy phenology. That even very closely located populations of the same species often flowered, blossomed at very distinct times. So it required a lot of riding around for the same populations to collect these fully developed flowers. So at the initial stage of the realization of the project, we had to cut down the kilometer distance. And what I can say from the field work experience, this was enough to already have some variability included, because yeah, even sometimes maybe populations located 500-600 meters away. One was at the edge of a forest, one was maybe in a country road, and they had 1 or 2 weeks differences in flowering. So yeah, so it was 5 kilometers.
It's quite often that nature verifies our expectations.
Okay. Any more questions, comments?
I don't know.
I'm happy it didn't happen this year, because everything
Yeah. Okay, so what's time? Yeah, we're almost on time. So I would end this session. Please thank again our speakers. And now we have time for coffee break. Please be back at 4:30.
as you know.
Thank you. And now we are going to start our session. Hello, everyone. My name is Marzena Guzicka. I work in Institute of Dendrology, and I have the pleasure of chairing this last session, during our conference, but of course not least. And 5 speeches are planned for this session. And let's start. First presentation, it will be presentation of Joanna Kijowska-Obert from our institute. And Asiu, floor is yours.
Thank you, Professor. Okay. Hello, everyone. My name is Joanna Kijowska-Obert, and I am a PhD student at the Institute of Dendrology, Polish Academy of Sciences. And today in my presentation I will talk about Proline as a component of metabolic pathways network in plant cells. We often hear about climate change, and we know that it causes shifts in the distribution of many plant species. However, it's rarely mentioned that these changes in distribution often originate from seeds. High temperatures and water deficits affect seed quality while they are still on the parent tree or while they are developing. Such conditions induce stress, which can lead to loss of seed viability and a decrease in germination ability. But on the other hand, stress can also induce acclimation and improve resistance to unfavorable environmental conditions. Populations identified as acclimatized to new climatic conditions would serve as source of seeds that do not lose viability during long-term storage. Such storage of seed resources is highly needed in forestry and nursery production. Therefore, through this identification, economic losses due to decreased germination capacity of stored seeds could be reduced. The efficiency of seedling production would increase, and these seedlings would be more resilient because they would come from populations identified as better adapted to current conditions. But how can we identify such populations? Biochemical markers may be helpful. Proline and other compounds related with its metabolism are promising indicators of seed and seedling quality. These substances are interconnected in a specific network of reactions, but changes in their levels or activities depend differently on the level of environmental stress. Therefore, determining the relationships between them and their response strength to stress is crucial to select the most reliable biochemical marker reflecting stress tolerance. Okay, why are we interested in Proline? It is a unique amino acid whose accumulation in cells increases in response to stress. Proline scavenges reactive oxygen species, ROS, which are abundant under stress conditions and damage cell membranes, leading to various disorders. It also protects the structure of proteins, stabilized membranes, building cellular organelles, and prevents cells from losing water due to its osmoregulatory properties. And models created as part of our meta-analysis of available literature data on proline accumulation in 3 seedlings under drought stress predicts, among other things, that the effect size depends on the duration of drought, while the effect is proline accumulation in response to drought. As you can see here, the optimum accumulation of proline occurs after approximately 70 days of drought, and this effect is non-linear. What could this turning point means? We have the theory. It could be the moment when acclimation occurs and the seedlings stop experiencing drought severely. Another model shows that the effect size depends on the seed category. There may be a relationship between seed category and the ability of resulting seedlings to tolerate drought stress. We have previously conducted studies on seeds of different categories. On Norway maple with seeds resistant to desiccation, therefore it's orthodox category, and on sycamore whose seeds are sensitive, so it's recalcitrant category. Indeed, recalcitrant seeds accumulate much more proline in response to increased temperature and decreased precipitation. We have also demonstrated that the effect size of proline accumulation due to drought stress is stronger in deciduous trees than in coniferous trees. As one might expect, the larger leaf surface of deciduous trees promotes faster water loss, and additionally the coniferous tree species included in the analysis produce seeds belonging to the orthodox category. Besides proline, enzymes involved in its metabolisms are interesting potential markers of drought resistant. Proline 5-carboxylate synthetase, we can call it P5CS, is responsible for proline biosynthesis. Its activity increases in response to increase in ROS levels, and proline dehydrogenase ProDH responsible for proline catabolism, and it is a mitochondrial enzyme. and its activity decreases in response to increase of ROS levels. One of our experiments showed that there's some tissue specificity. P5CS activity was higher in roots than in leaves, unlike ProDH activity. Moreover, the increase in P5CS activity was correlated with the hydrogen peroxide accumulation. And the strongest correlation was observed between pro-DH activity and soil moisture level. And it was correlation, negative correlation. And it is very important for us because pro-DH seems to be really a promising marker of drought tolerance. Okay, we have already discussed potential stress tolerance markers belonging to the proline metabolism cycle. However, we also have other candidates for this position. And it's noteworthy that all these candidates are interconnected and work together like a set of gears of two wheels, allowing the plant to defend itself against the consequences of drought. We aim to further determine the relationships between them and their response strength to stress, to select the most reliable marker. And one example could be nitric oxide. Nitric oxide is a small but important redox signaling molecule that in plants is involved in a diverse range of physiological processes, including germination, development, flowering, and NO has emerged as an important signaling molecule in plants, activating ROS scavenging enzymes under abiotic stresses. Plants grown under water-limited conditions undergo various adaptive mechanisms to reduce stress. Most of these adaptations resulting from limited water conditions involve NO. Its role in drought tolerance is enhancing the antioxidant systems, proline and osmolyte metabolism, and/or reducing ROS levels via antioxidant enzyme activity, and can reduce superoxide anions and hydrogen peroxide when they reach toxic levels. At molecular level, NO plays a multi-dimensional role in drought stress. It has been shown that in response to drought, plants exhibit DNA methylation, and these cellular changes are affected by NO. But plants can defend against oxidative stress also through the ascorbate glutathione cycle by increasing the activities of ascorbate peroxidase and other enzymes involved in this cycle to scavenge hydrogen peroxide. Under the influence of NO, the expression of antioxidant enzymes involved in the ascorbate glutathione cycle increases, which in turn helps alleviate oxidative damage caused by ROS. The activity of ascorbate glutathione cycle enzymes can be maintained by Proline, thereby increasing stress tolerance, and upregulation of both P5CS and APX activity may be involved in drought tolerance mechanisms. New evidences suggest that proline biosynthesis enzymes interact with redox proteins So an important future pursuit will be to identify other interacting partners of Proline metabolic enzymes to uncover novel regulatory and signaling networks of cellular stress response. So what about future possibilities and applications of Proline as a component of a metabolic pathway network? We can identify populations producing high-quality seeds, and it will make seed storage more effective because of limiting the loss of seed viability. Then seedlings will be more stress resistant. And we will observe increase of survival rate of regeneration in forests. Reducing economic losses caused by the loss of seed viability during storage and by the mortality of forest regenerations will make subsequent generations of trees in forests more resistant to new climatic conditions. Thank you very much for your attention.
Thank you very much for a very interesting presentation. And our next speaker will be Anna Skorti from Agricultural University of Athens. And Anna visited our institute a few months ago, and she is a friend of our institute.
Thank you. Thank you very much.
I think I can say this, and I'm inviting you for showing us your results.
Well, good afternoon. I'm Anna Skorti. I'm a postdoctoral researcher at the Laboratory of Agricultural Zoology and Entomology, Department of Crop Science at the Agricultural University of Athens in Greece. I'm going to present you our research work with the title Monitoring and Management of Limandria dispar life stages in southern Greece. Limandria dispar, with a common name Gypsy moth or spongy moth, is the new common name, is one of the most destructive insect species, reaching third in the list of the most economically harmful species worldwide. It is a notorious invasive polyphagous pest, causing a widespread loss of leaves in forests in Europe, Asia, North America, and parts of Africa. And it is also considered very harmful to orchards and urban environments. The lifespan of gypsy moth adult is about a week, and adults do not fly. Eggs are deposited in clusters directly on the bark or branches of host trees and in late summer. Eggs clusters have teardrop shaped. Their color is yellowish to brownish, felt-like in appearance, and turn lighter in color with age. Normally, all individual eggs are hidden by the hairs of the female abdomen. In addition to host plants, females of the European subspecies have little to no selectivity during oviposition, and they can deposit the eggs masses on available substrates like rocks, walls, or other immovable objects. On this slide, we see the appearance of the first instar larvae. It is about 3 millimeter long. And here is the appearance of the last instars, which is sometimes up to 90 millimeter long. And it also has 2 characteristic rows of 6 red spots, as you can see. Pupae are teardrop shaped without a silky cocoon. Males are usually smaller than female and have brown silk threads that hold the nymphs to the tree trunk. Limandria dispar has 1 generation per year and hibernates as egg. from September to early May approximately. After hatching, the larvae feed on the foliage of the host plants for a period of 6 to 8 weeks. The 80% of defoliation is conducted by the 5th and 6th instar larvae. In Central Europe, the larvae begin to feed in late April and continue for up to 10 weeks until they pupate in late summer to in late June to early July. The adults appear around mid-July until the end of August, depending on the weather conditions. Lymandria dispar is a serious insect pest species in southern and central Europe. It was intentionally introduced to the east coast of North America from Western Union in 1869 for silk production, but a small number of individuals escaped the laboratory, and since then it has become a much more serious pest in the United States than in its native regions. The spread of the gypsy moth in Europe has been recorded in the database of the European and Mediterranean Plant Protection Organization, EPO. According to this database, this species is present in more than 20 countries, including Poland. Lymandria dispar has the ability to be carried by the wind on silk threads in long distances, or by depositing the eggs on objects that can change positions, such as firewoods, Christmas trees, boat, cargo container, or trailers. Gypsy moth is a polyphagous species, as it can feed on a wide range of hosts, belonging to 400 to 600 plant species, with a preference of species within the genus Quercus. In Europe, the preferred hosts vary by region, but include species of Quercus, Carpinus, Alnus, Prunus, Populus, and Robinia. Defoliation by gypsy moth larvae can cause tree biomass losses up to 70%, while cumulative effect can result from consecutive defoliations. Defoliation causes feeding problems of grazing animals and birds. Outbreaks are periodic, every 5 to 10 years. From an economic perspective, the gypsy moth can massively affect timberlands and agricultural products, with high yearly costs on quarantine products. More, they impact the aesthetic of forests and property values in urban areas, imposing high costs. It is also burdens health systems and may also negatively affect the tourism. In addition, larvae of this insect is converted with hairs, the setae, which are full of toxins that can cause allergic reactions in humans and animals. More specifically, they can cause eye irritation, dermatitis, rhinitis, dyspnea, swelling, shortness of breath after contact or inhalation. First instars larvae considered more allergenic than other stages. On this slide, we see some allergic reactions. About the control of Lymandria dispar, in biological control, there are more than 165 natural enemies. Control can also include bioinsecticide or pathogens, attractants such as sex pheromone traps, and chemicals. And now the aim of this present research project is the use of smart traps and pheromone traps to monitor and control of Lymandria dispar in Greece. This research conducted by the Agricultural University of Athens as part of the European research program Life Mer, with the title Using Smart Traps and Pheromones to Control the Gypsy Moth Eco-Friendly and Control in Practice, which has additional participants Slovenia and Spain. The experiments were conducted in Ilia, a prefecture located in the southwestern part of Greece. The first part of the project included the capture and control of Lymandria dispar larvae through trunk traps. The purpose was to compare the effectiveness of three different types of trunk traps, two commercial, trap number 1 and trap number 2, which has collecting bugs, and a prototype trap number 3, which has the use of glue. These traps installed on 17th of March 2023. And their effectiveness was being monitored weekly until July of 2023. In the results, according to the diagram, the highest number of captures was observed in trap number 3, the prototype trap, showing a significant difference in the number of captures compared to the other traps. Next project part included the capture and monitoring of adults Lymantria dispar through 6 different prototype pheromone traps and a commercial pheromone trap. The project included 2 experimental protocols. The purpose of the first protocol was to compare the effectiveness of 3 different pheromone traps. The G trap, which was a commercial trap, with 2 prototype traps, the Igimer 1 with the red color without insecticide, and the Igimer 2, which is the modified Igimer 1 with brown color and lambda cichalothrin insecticide. On this slide, we can see the experimental design. The distance between the traps is in the same block was approximately 70 to 100 meters, and the distance between the blocks were approximately 150 meters. The purpose of the second protocol was to compare the effectiveness of 4 different pheromone traps, the G trap with 3 prototype traps, the Igimer 2, the Igimer 3, which is the modified Igimer 1 with the brown color without the insecticide, and the Igimer 4, which is the modified Igimer 1 with brown color and deltametrin insecticide. On this slide, we can see the experimental design. Distances are the same as before. The pheromone trap installed in 2 of June 2023, and their effectiveness was being monitored weekly from June to September 2023. The results, in the diagram, we see some differences in the number of captures, with the maximum occurring in G-trap for the first protocol, while the second protocol, the maximum number of captures is presented in Igimer 3 trap, followed by Igimer 4, while significantly fewer captures were presented in G-trap. Next project included mating disruption. Mating disruption is a novel, promising technique for pest control. Artificial female is released to compete with the pheromone produced by calling females. Mating disruption has been successfully marketed for a very wide range of moth species of economical importance. With this method, mating may be delayed, which contributes to reduce fecundity and egg fertilization. On this slide, we see the experimental design. In the right picture, we see the areas where the pheromone gel has been applied to the tree trunks. We also see the traps that were used to test the effectiveness of this method. The mating disruption gel application took place on Friday, 2 of June 2023, and the traps installed the same day. The effectiveness was being monitored weekly from June to September 2023. And the results showed the success of the MD application, the mating disruption application. We can see the low number of captures in the application area compared to the control area. And in this diagram, we see the average of the captures for the entire experimental period. The last project for 2023 included the monitoring of Lymantria dispar through electronic traps. On this slide, we can see the design for any trap. The trap is automatically charged by solar chargers. The installation took place in June and July 2023. With electronic traps, we have the opportunity to check at any moment the weather conditions of the environment and how the insects is affected by them. In addition, the captures are recorded daily, 3 times a day, through a camera and saved in an application which is available at any time by the users. More information and details about the program's action you can also find on our social media pages. And more, we are guest editors in the special issue of the international journal Insects. In this special issue, 11 papers have already been published and several others are under consideration. So we welcome contributions dealing with agricultural and forest ecosystems about pest management. Insects is Q1 in the entomology category, has impact fact 3, is fast in the review process, high standard journal, high quality and high visibility. That's it. After the finish of this session, you will have the chance to see these pheromone traps device live outside. Thank you very much.
Thank you, Anna, for a nice presentation. And now I'm inviting David Gerna. I'm not sure if I correctly pronounce your name. I'm sorry if not. David works in Kew Garden in UK and show us some aspect of cryopreservation of Quercus robur.
I'm sorry, the mouse is not working. Great. So thank you very much for the kind introduction and for the opportunity to present some preliminary results on this pilot study that I'm conducting at Kew as a research fellow. So let's crack on with how we can help plants maximize their survival by investigating if the cell cycle influences cryopreservation. We know that plants are under threat due to current global change, and the rates of disappearance of species is particularly fast in this period. So we have an opportunity of protecting plants by safeguarding plant diversity through seed banking under dry and cold conditions. But this is not an option for all seeds, because we have recalcitrant species, as we heard before, which cannot withstand the desiccation and freezing. And we know that worldwide at least 50% of recalcitrant seeded species is classified as endangered and vulnerable, and this issue is particularly severe for tree species, including the genus Quercus, which is the initial target of this pilot study. So cryopreservation is the storage of plant material at liquid nitrogen temperature, so I'm talking about minus 180 or 19 Celsius. And for oaks, we used a cryopreservation protocol which consists of several steps, starting from the excision of the embryonic axis, followed by partial and fast desiccation, plunging and storage into liquid nitrogen. And after storage, we have a rapid reworming completed by in vitro regeneration of seedlings, which is the final step determining the success of cryopreservation. In Quercus robur, which is the initial targeted species, or in this pilot study, the percentage of embryonic axis survival is pretty low, and values are further curtailed if we consider the percentage of seedlings with a normal phenotype, with both root and shoot. And we know that in this species, one main cause of damage is related to desiccation. However, the influence of desiccation on the cell cycle and in general plant growth remains a big unknown. So the cell cycle is a process typical of proliferating cells, which consists of two main phases of DNA synthesis and cell division, or mitosis, separated by two gap phases of cellular growth and separation, so G1 for DNA synthesis and G2 for mitosis. And we know from desiccation-tolerant species that the progress of the cell cycle, or a higher ratio between G2 and G1 cells, has been related to a more synchronous germination and higher seed vigor. So we hypothesize that the success of cryopreservation of recalcitrant species, seeded species, may be related to, maybe improved by synchronizing cells at the G1 phase of the cell cycle, when we have a lower DNA content, and so cells are supposed to be more resistant to cryo damage based on animal models. Furthermore, embryonic axes are exposed to oxidative stress throughout the steps of cryopreservation, starting from excision and partial fast desiccation, but also during thawing and in vitro regeneration of seedlings. We are initially focusing on the desiccation response, and we know that blotting excised embryonic axes on solutions of antioxidant is beneficial for survival. However, the mechanisms behind these effects are less clear, and we assume that they may relate to the cell cycle, because we have two checkpoints at the G1 and the G2 phase, which appears from other studies to be redox sensitive, which means that they respond to pro-oxidants like reactive oxygen species within certain levels and antioxidants, that they both established redox cycles within the cell cycle. Furthermore, the cell cycle also requires a rearrangement of the membranes, and this goes along with changes in the structural composition of lipids. So we are also looking into this component. And our overarching aim overall is to try to understand and elucidate the cellular and redox mechanism of plant desiccation tolerance to leverage the survival and ex situ conservation, particularly of species which are difficult to store, short-lived or recalcitrant, let's say exceptional species, as was mentioned yesterday. So just very quickly, some background information on the anatomy of Quercus robus seeds. So we have two big fleshy cotyledons with storage tissue, and then an embryonic axis, which is made of a radicle and a plumule. And we use the embryonic axis in our cryopreservation protocol after chopping out the cotyledons, because they are extremely bulky and they would lead immediately to ice crystal damage and injury, besides slowing down desiccation and freezing. In more detail, here we have a cross-section of a seed, and you can see the two white areas at the streams. They are, of course, the root and the shoot apical meristems, which are connected by an embryogenic vascular tissue, which is the procambio. And we know that it is important for transferring nutrients across the two meristems from some 3D reconstructions. So in fresh seeds of Quercus robur, the embryonic axis has a significantly higher water content than the cotyledon. But to quantify the amount of unbound freezable water, which is responsible for ice injury, we ran some differential calorimetry analysis in both seed structures, so the cotyledons on top and then on the bottom the embryonic axis, in both fresh seeds and dry to very low moisture content, as you can see from the bottom. So the samples were cooled down to minus 150 degrees and then warmed up to about 40 degrees. And you can see that we have peaks only in the samples at high moisture content. So this can be related to the unbound freezable water. And particularly the areas which are encompassed by the peaks are proportional to this. They are a measure of the melting enthalpy of freezable water, which was indeed significantly higher in the embryonic axis than in the cotyledons, which indicates the need of at least desiccate partially the embryonic axis before any freezing to avoid ice crystal formation. So in this is indeed to test our hypothesis. We work on a protocol which starts with the excision of the embryonic axis, followed by the blotting in a solution of ascorbic acids, and then we conduct like a physical drying very fast, much faster than under a laminar flow. And this is a no-made device where we use like a sealed jars. At the bottom you can see some regenerated, very dry silica gel, and then in the middle there's a fan with a mesh which is providing like a flow of air to desiccate the seeds, and then we score the survival in vitro. Of course we need to do this in vitro because the storage tissue is gone, so the embryonic axis needs nutrients, and we monitor the survival at different intervals to build seed survival curves of desiccation. And we distinguish between the responses of the root apex and the shoot apex, which are shown here in these graphs on the top in pink and in green, respectively. So regardless of desiccation, we generally have higher frequency of radical regeneration rather than shoot formation from the apex. And what we observe is that during flash drying, the shoot apex was drying much faster and to significantly lower water contents. And regardless, both apexes responded with a dramatic loss in the survivor already from the first desiccation interval, as you can see on the right, and therefore we implemented the protocol with two initial cryoprotection steps, which involve 5 and 10% glycerol, each for 1 hour, and the outcome is shown there at the bottom in the yellow frame. So in terms of water content, the changes occur more gradually and at a similar rate in both apices, and importantly, the water content of the shoot apex was consistently lower than in the root apex, and this has had influential consequences on the survival, positive, because you can see that the survival was maintained to high rates up to 3 hours from the onset of the flash drying. So this is very much still work in progress, but we are using flow cytometry to quantify the proportion of cells in the G1 and the G2 phase of the cell cycle using individual embryonic axes for each of the cryopreservation and the cryoprotection steps. And we are also looking at the effect of the influence of exogenous pro-oxidants like hydrogen peroxide, which is stable for quite some time at low temperature, and then antioxidants like ascorbic acids and glutathione. So on these slides, you can see different flow histograms from the analysis of whole embryonic axis from fresh seeds, so just the very first step of the protocol from the previous slide. They were processed with an internal standard for relative quantification, which is denoted by the IS peak in each of the 5 flow histograms, and each of them refer to a different embryonic axis. And based on the genome size of the species, we know that the first clear peak that you can see from the left is related to cells in the G1 phase of the cell cycle, but we were not able to detect a peak of cells in the G2 stage under these conditions. And this was true also when we considered cleanly excised root and shoot apical meristems from the embryonic axis. Again, there was no detectable clear G2 peak. So the conclusion for the moment is that at least in fresh seeds after maturation, after shedding, most embryonic and meristematic cells of the embryonic cycle are almost perfectly synchronized in the G1 phase, and there is no need for an initial synchronization. However, to show that we are able to detect peak in the G2 phase of the cell cycle, we use hydroxyurea, which is an inhibitor of DNA synthesis, causing an accumulation of cells with G1 cell, an accumulation of cells in the G1 phase of the cell cycle, and we screened a range of concentrations for the effect on seed germination, and indeed we found that a couple of concentrations among those tested were able to restrict the growth of radicle and therefore slowing down germination, as you can see from the rectangle at the bottom. And then from seeds with different radical lengths, we excised the root apical meristem, and indeed you can see that a small G2 peak, but detectable, is appearing at the expected position after the G1 peak. And this peak was disappearing when we exposed the same explants to the inhibitory concentrations of hydroxyurea. So the G2 peak went away. And we suggest that in this species, radical protrusion and growth is mostly based on cell expansion and differentiation rather than cell division and mitosis. Again, we have a lot of interest into looking into the response of membrane lipids to desiccation. And to do this, we have been running since the last 3 weeks or something like this, high throughput analysis of the lipidome, and we are using a stepwise approach. So after the extraction, we conduct a direct infusion mass spectrometry without any chromatographic separation, and this enables to reliably detect and confirm the molecular structures of a various range of lipids, including sterols, triglycerides, phospholipids and constituent fatty acids. And then after this step, we can further confirm the identity of our molecules using liquid chromatography coupled to mass spectrometry, which is a more targeted method to better reveal the nature of lipids and quantify the lipids changing most prominently across treatments. As I said, this is very much work in progress, and I hope to be able to report on this soon, next time we meet. And finally, another important aspect of Quercus robur, I think, is related to in vitro culturing from the embryonic axis, regardless of desiccation and freezing, which I find quite challenging. So we decided to test the effects after excision of higher than usual concentrations of ascorbic acids and then also glutathione and hydrogen peroxide, and look into the seedling phenotypes after 5 weeks of growth on WPM medium. So in the control, as I mentioned earlier, we typically have a more frequent growth from the radicle than from the shoot. It's about 4-fold mold, and after the redox treatments, we could observe an increase of about 3-fold in the percentage of seedlings with a normal phenotype, so with both root and shoot clearly growing. So to conclude, some take-home messages from this presentation. One is that the survival of desiccation in Quercus robur and many other recalcitrant species is still a critical step, which limits the successful application of cryopreservation, and we are aiming to identify molecular markers of desiccation sensitivity, looking into the cell cycle and into the lipidome. Second, when working with the embryonic axis, it's important to distinguish between the responses to the desiccation by root and shoot. In this case, in this species, they displayed differential responses to desiccation, and we were able to improve survival rates with some initial cryoprotection treatments before flash drying. And finally, based on the last slide that I show you on the in vitro culture, we assume that the modulation of the extracellular redox environment during the in vitro growth dependent on the independent of cryopreservation is another critical aspect that needs to be considered for future direction to enhance the survival and growth of plants in vitro. And with this, I thank you very much for your attention. I acknowledge the financial support by the Ministry of Science and Higher Education of Polish and the invitation and all the people who are supporting the delivery of this project. Thank you very much.
Thank you, Davide. It was a very interesting presentation with a lot of very nice and totally new results. Congratulations, Davide. And now it's time for Magdalena Terlecka, PhD student from our institute. And Magdalena give us some information about the genetic and clonal structure of the Populus alba.
Okay, so good afternoon to everyone. My name is Magdalena Terlecka, and I'm pleased to tell you about the genetic and clonal structure of the white poplar. So this is a topic from a bigger project, which is ecological consequences of clonality and dioiciousness exemplified with Populus alba. Okay, so at first a few words about the species. So white poplar is a dioecious and clonal species from the Salicaceae family, and it could be commonly found in river valleys and floodplains. And very characteristic, like you can see on this photo, in this species are white bark with something like rhomboidal squares on the bark, and also white downside of the leaves. And the pollen and seeds are dispersed by the wind. Okay, so white poplar is part of the biologically diverse poplar riparian forests, which are characterized by unique living conditions. But due to drainage works and the river regulation, riparian forests become more rare in Europe. And the result of that is the spatial isolation of white poplar population, and that limits the natural regeneration of the species. So actually we need to understand the genetic resources of white poplar, including its variability and factors that influence this variability. Additionally, it's also important to consider the consequences of clonality in combination with the dioecious nature of white poplar, and that knowledge is crucial to effective protective of this area and white poplar. Okay, so I will talk about two populations of white poplar. So Rogalniński Landscape Park here and Dębiński Forest. So on the map you can see distribution of the individuals of white poplar. So in red are the female one, and in blue the male one. So each of population has more than 500 individuals. Okay, to recognize gender of okay, so the recognition of gender is based on the inflorescences, like you can see on this photos. Male one have red one inflorescences and female green one. So poplar blooms at the end of March and beginning of April, and normally in most cases inflorescences occur at the height of 15-20 meters high, so mostly binoculars are necessary to determine the gender. To recognize clones and of course singletons, I make a few steps. At first I collected leaves from each tree and prepared them to isolation. Like you can see on this movie, I used liquid nitrogen and greened the leaves. And after that, I make a DNA isolation and also 4 multiplex PCR reaction. And after that, I analyze the DNA fragment length on the genetic analyzer, and that gives me information about genotype of each individual of each tree. For now, I analyze the genotypes using 2 packages, AirClone and poplar in R, but yeah, I hope I will use more packages for the genetic population. At total, I use 20 microsatellite markers for those 4 multiplex reaction, and the multiplex reactions were matched so that the markers, microsatellite markers, have similar reaction kinetics, like the same temperature of annealing. Okay, to to check if that number, that size of microsatellite markers will be enough, I check it with genotype accumulation curve using poplar packages. Yeah, like you can see, 16 microsatellite markers will be enough to describe all multilocus genotype in both population. Okay, so let's look at the clonal structure of both populations. So at first, the Rogalski landscape park, and it looks like that. So 501 individuals were included in the genetic analysis, and among these three, we defined 174 multilocus genotype, and inside of that, 96 turned out to be singletons. So they did not produce ramets, and inside the singletons we have 35 males and 61 females. And the remaining 78 turned out to be cloned, so they produce clonally. And inside of that we have 42 males and 36 females. For total, we have 196 ramets and 209 ramets respectively. Okay, so the situation in the Miski forest is quite similar, but we have a bigger population, so we have include 567 individuals. We have less multilocus genotype, and this is how looks the singletons and clones inside, and we have more ramets in that case. The number of observed multilocus genotype is equivalent of genotyping richness. In that case, so we have two populations of white poplar, and we want to compare them. More appropriate comparison is using the expected multilocus genotype, but still we see that the Rogalski landscape park is more genotyping rich than the Miski forest. Okay, some numbers. So, like you can see, the Miski forest is bigger, but inside the population we have less Alders and less unique genotypes. If we look at the clonal diversity index, it's quite low, and the higher the index is, the bigger is the genetic diversity inside the population. So in that case, we have quite low genetic diversity, and we have a comparison for that in observed and expected heterozygity. Quite interesting is if we look at the pillow index, which told us about the clonal architecture. So if it's near 1, that mentioned that in the spatial area of the multilocus genotype occur only ramets which are from this genotype. Sorry, stress. So in that case, we could see that the population, the spatial area of each genotype are very dense, because we have only ramets from a given genotype in the area. And the inbred coefficient is quite okay in the Miski forest, but it could be afraid in Rogalski landscape park because it's almost more than 10% of inbred. Okay, to conclude, in the case of both population, clonal reproduction turned out to be the dominant method of reproduction. And the result of that is low clonal diversity, because ramets of given clone appear and the same genotype are repeated and repeated. And because of that, population regeneration is slowed, because responsible for the genetic variability is sexual reproduction, and it's limited because of dominance of clonal reproduction. Okay, so information about genetic resources will answer the question about the condition of a given population, and we could make some predictions. Will that population survive in the face of climate changes, or will it not? So in our case, the genetic diversity is quite low, so it could be threatening. Okay, so like I said, this is a topic from a bigger project, so I hope that next time I could tell you more in wider context about genetics, but also about ecological context and dendrochronology. So I want to thank my supervisor, so Professor Białogorszko and Dr. Katarzyna Stankiewicz for help in field and laboratory. Thank you for your attention.
Thank you, Magdalena. It was a very interesting speech. And now I'm inviting Dominika Thiem. It will be the last presentation during this session, last but of course not least. Dominika is PhD student work at University in Toruń. Yes?
Yes, it's true.
Nicolaus Copernicus University in Toruń.
Good morning, and maybe good afternoon, I should say now. Yes, because it's the end of our conference. It's really nice to see you all here. And my name is Dominika Thiem, and I am representing Nicolaus Copernicus University. I still look young, so everybody thinks that I am a PhD student, but it's not true. So today I would like to tell you a really short story about the Alnus glutinosa. And during this conference was a lot of talk about the climate changes. And I would like again stress that climate is changing on our eyes, and we can see directly that air temperature and deficiency of water is still decreasing. For this reason, we have a new problem, the soil salinity, and so far it's have reached more than 900 million hectares over the world. One of the solutions for this can be tree planting, because trees have the significant ability to decrease the water table and thereby reverse some process of salinization. Additionally, the trees can form some symbiotic association with ectomycorrhizal fungi that can help to protect the trees against this saline stress. And this can be by balancing water relations, by the improvement of nutrients, by alleviating sodium ion uptakes, by reduction of osmotic stress, or by decreasing reactive oxygen species level. And while the information about the microbiomes of crop plants are really good described in the literature, really little is known about the trees under saline stress condition. For this reason, I started to broaden this knowledge during my PhD, and I am continuing it now. I am working with Alnus glutinosa. This tree is specific for two reasons. One of those is that this tree can tolerate high different abiotic stresses, example salinity, and can form really specific microbiome in the roots, because can associate it with arbuscular fungi, with ectomycorrhizal fungi, and with nitrogen-fixing bacteria at the same time. And on the basis of my PhD, I checked that in the roots we can observe other endophytic microorganisms, different bacteria and fungi. However, ectomycorrhizal fungi seem to be the most important for the survival of these trees. Why? Because we check it in pot experiment and we know that is it that. And this ectomycorrhiza in adult trees, it's dominated. For this reason that we don't know, we know only something about the microbiomes of these trees, the main objective of this study that I show you was to characterize the fungal microbiome of absorptive roots and the rhizosphere, but with the special attention on ectomycorrhizal fungi. I hypothesized that fungal diversity can be higher in the rhizosphere than in the black alder roots because of the diversity in the soil, and the diversity of ectomycorrhizal fungi will be more often found in the root rhiberries than in the soil samples. The second hypothesis that I put is that maybe two factors that we analyze can be significant, change the relative abundance of ectomycorrhizal fungi by decreasing or decreasing the sum general and the abundance. And the third one, that is exactly connected with the negative effect of the salinity, that an increase in soil salinity can negatively impact on sub-parameters that we measure. So the production of the roots, the diversity of ectomycorrhiza morphotypes, and the number of ectomycorrhiza root tips. Roots with the surrounded soil were sampled in 3 tested sites that were located in northern Poland. The control site was located in Szczuczyn, really nice name in Poland. And 2 saline sites were located in Słonawy, but in different district areas. The distance between these 2 sites was 8 kilometers here, and at each site we distinguished the same area. We sampled the samples from 3 plots, and randomly we selected 9 trees, so in total we had 27. It can be maybe little, but in case of the analysis that I will show you, it's a lot. The tubes of the mini rhizotron that you can see here, we put to the surface of the soil in 2017, and I did this experiment in 2018, but I will show you the information about the one growing season in 2019. The samples of roots and rhizosphere were taken in 3 seasons and 3 months, April, July, and September, that I assigned to the spring, summer, and fall. The bulk soil were used to receive the physicochemical parameters. The rhizosphere soil were separated from the roots were sterile, and these 2 sources were used to preparation of ITS libraries that were sequenced by me using Illumina MySeq. But the second part was about the mini rhizotron cameras, and I did in every end of the month the pictures, so I traveled to the sites and I did the pictures in every month, and this information will use to the analysis of the production of absorptive roots, but the information about the ectomycorrhiza I did only for April, July, and September because of time. So now we can jump to the result part. This table is presented soil conditions. So here you can see that electrical conductivity that is connected with the level of the salinity was the highest at SH site and lower as at SL. So we confirm this, but pH was the highest at SS. NS site and SL site characterized by the highest level of magnesium, whereas the SL site characterized by higher level of pH and calcium. And the salinity at this SH site was positively correlated with the level of sodium and chloride ions in this soil. Now we can check the first hypothesis that I talked about. On the left side, you can see the box plot with the results about the alpha diversity. And you can see directly that in rhizosphere soil, it's exactly higher alpha diversity compared to the roots. When we check the information about the relative abundance, ASV that was assigned to Telephora, Acephala, and Phyalocephala were more frequent in the roots compared to the soil, whereas Podilla, Mortirella, and Alternaria were more frequent in the soil compared to the roots. Next data about the genera I shared on the basis of the fungal trait database that you can receive some information about the ecological function of the fungi. And you can share an example for the primary lifestyle, and I did it. And I received the information about the ectomycorrhizal fungi, different group of subtrophs, and plant pathogens. And I focused, of course, on the ectomycorrhizal fungi so far. And I found that during the spring and summer, so this hotter time during the growing season, so here, we observed that ectomycorrhizal fungi have higher relative abundance at saline sites compared to the non-saline, where the opposite tendency was observed in the fall season. So here. And I tried to understand why is it like this. So we checked the rhizosphere, of course, and maybe that rhizosphere is the source of this ectomycorrhizal fungi. Maybe there will be the same tendency. But this tendency is observed only for SL site, so maybe it's some additional factor that we need to find to understand this. Among these ectomycorrhizal genera, we identified 11 different genera, but among them only 4 was dominated. And here you can see that Telephora species, both in roots and in the soil, always was higher abundance at as a saline site compared to the non-saline, whereas when we look for the genera Tomentella, Lactarius, and Rusula, you can see that we can observe higher relative abundance at NS site compared to the saline ones. Additionally, in the rhizosphere soil, we found a lot of asphals connected with the Inocybe species, but the relative abundance was high only at SH site, what you can see here, and it was observed only in the rhizosphere soil. Now I will go to the negative impact of salinity on the almus. And here you can see the information about the average number of absorptive roots. So here it's 10 times more roots compared to the saline sites. When we look for this bar plot from 2018 to 2019 to the end to September, we can see the peak here and 2.5 higher production of the roots at non-saline site, but in the saline site we can observe similar production. Here is the picture from the mini rhizotron. I wanted to show you this. We published this on the end of the last year because it's, in my opinion, really great. And here we can see non-mycorrhizal roots, alnus. You can see the red tip on the end. But here are some examples of the ectomycorrhizal fungi that we assign to some genera, but of course it's the probable identification because it's in situ. So we cannot take it, cut, and check what is inside. Now in this year, in 2019, we did a calculation of ectomycorrhizal root tips, and we received negative effect on them on all of the seasons. However, in summer and in fall, we didn't observe the differences between SH site and NS site, maybe because of broad range of the values that we receive. And here you can see some examples of the picture. This is non-ectomycorrhiza, and this is different morphotypes. And I am doing the assigning this to the genera. But the morphotypes for the NS, it's only 5, but for the saline site, it's 3 or 4, depends on the tubes that we analyzed. So to conclude, I would like to say that microbiome of alnus roots show the lower alpha diversity in the roots than in the rhizosphere soil. Genus Tomentella, Telephora, Rusula, and Lactarius belong to the 4 main genera that we observed in absorptive roots, both in saline and non-saline conditions. More ectomycorrhizal fungi can be recruited to alnus roots from soil during spring and summer than in the fall at saline sites compared to the non-saline, so it depends on the season. And that the salinity decreases the number of absorptive roots, abundance of genera, and number of ectomycorrhizal morphotypes and quantity of ectomycorrhizal root tips. And on the end, I would like to recommend you to mix different techniques, because the mini rhizotherm thing that is maybe quite new with the new generation sequencing, and we can use it really good to analyze the ectomycorrhizal community. Thank you very much for your attention, and thank you for all of the co-authors and for the organizers of the conference.
Thank you very much, Dominika. I apologize for my mistake in introduction you. And now we have time for questions. Unfortunately, our first speaker, Asia Kijowska-Oberts, had to leave us, and we have no possibility to ask her questions. But if you are interested in her results, we can contact with her by email, and this email there is in our book of abstract. And now I'm opening discussion. Okay. First question. 2.
Yeah, thank you. To the last speaker. Maybe I've missed the information, but I would just wanted to know if you mentioned the region that you amplified, talking about the molecular part of the experiment, and which primers. And also another question, why did you choose fungal traits as a database, preferably to another one?
So maybe first about the primers. I designed it by myself during the PhD, and I tried to catch the different fungi, so receive broad range of the fungal community to see information about the ectomycorrhizal fungi and arbuscular fungi, and I'm using it longer time, so it's not the universal primer, but I am doing the designing primer for this genera that I found now, and I try to do qPCR to check how much is it in the samples. And the second one was about the saline database, okay, about the fungal traits. It's really good because the person who described it, it's the excellent person in analysis of ectomycorrhizal fungi. And I check all of the data that was there, and in my opinion, it was really good assign. So because of that, I use it. And I recommend you if you want to use it.
Thank you, Dominika. Yes, we have next question.
First, I'll also ask Dominika. So you mentioned at the beginning about the ability of alders to fix the atmospheric nitrogen. Do you think that the increased salinity will somehow affect it? And the second one is about Phytophthora. Do you think that salinity will somehow influence those infections?
I show you the information about the microbiome, but we did, of course, the bacterial microbiome additionally, and it's affected by salinity too. But in case of Frankia, I see that we can observe more nodules on alnos that are growing under saline stress conditions. So maybe under these conditions, the tree needs more nitrogen. So maybe it's, it's helpful to survive under these conditions. Good, I understand it. Yeah. I read something about this, that alnos, it's yes, it's Phytophthora, but I don't have really idea about this.
Are there any questions?
If I understood you correctly, you couldn't cut a part of a root to check what fungi are inside. Why couldn't you do this?
We try to use only lead-to material and not disturb the trees. So we take only one small falcon with the roots to sterilize them and to use. And the idea is to analyze these trees without the disturbance of this.
But you used only 3 trees in each place. You could pick some from a 4th one, just for curiosity.
It was 3 trees per plot, so 9 for each test plot. Yes, each site. The number was because I received some money from National Science Center, and to the mini rhizotron you need to buy special tubes that I ordered for the United States, and I need to buy for everything. So we calculated how much we can buy of these tubes to do this analysis, and we calculated that 27, it's enough money to do it. So it's 9 replicates per each. Yes. So maybe for the future, if I will receive bigger grant, I will put more tubes.
It's just a comment addressed to David and Joanna. I don't know if she's here. Okay. Actually, currently we are working on the conservation of seeds, tropical seeds in Africa. We have a grant, and we must test around 30 species to know we can conserve the seed for how long. And we have the ability to study the physical aspect. If you are interested in molecular, genetics, and chemistry aspect, you are welcome.
Yeah, thank you very much. Sometimes we have this, with Q, it's these bilateral agreements, so you're feel free to get in touch with me, but it's not dependent of me, but we can look into it. Do you know if these species are, what's the storage behavior? Do you know anything about? Are they, okay, so, but we can, okay, thank you.
Hania?
So as a representative of Asia's lab, I invite you to talk about it at the barbecue.
Are there any questions? Yes, please.
So, Magdalena, in Estonia we have one aspen species that is native one, that is Populus tremula. And this tree is really important, specifically the old trees that have already holes in the wood, because they are home to the critically endangered flying squirrels, or maybe it's the right word is gliding squirrel, I don't know. So my question is, do you have any of those in Poland?
Populus tremula, do we have in Poland? No. No, we don't have any. Okay, so yeah, we don't have.
I have a question to David. Do you think about some microscopic observation? I think, for example, structure of nucleus or location of lipid bodies in cell.
Yeah, that's indeed very interesting. I think in this species there was some already some study from the late 90s from Patricia Birch, possibly, but it's of course something very interesting to do. Wow, this is just a pilot study, but I guess can be developed. So I think it's very much important when we look to the into the desiccation and freezing responses to have indeed in detailed ultrastructural studies, looking at the cellular compartmentalization and location and eventually so that's for sure something ultrastructure. So thank you.
Are there any questions? Eva?
I have a question to Davide, because I think that it's very interesting to use oxidants and antioxidants in the protocol for cryopreservation. But I have a question. Did you check which of these molecules are the most efficient in increasing the seed survival or seedling growth? Do you have some observation?
Yes, so the reason for choosing the concentration that I show you and testing on the embryonic axis is based on some preliminary screening on germination, where indeed we had an effect. I would like to repeat because I think it's very much dependent on the year and on the season, but we have like a slight speeding up of germination by the hydrogen peroxide, whereas the two antioxidants, there was a tendency, especially at the first stage of germination, to hold germination back. So I think there is this something very interesting to look at about. So this, and indeed, we selected the concentration which were affecting the germination of acorns, like slowing down or spinning up, and then assess that they were not toxic, for example, for the embryonic axis. And it was not the case, but when we worked only on the embryonic axis only, it was not clear to distinguish in vitro, if they were slowing down or speeding up. Was just a matter, because the evaluation there is a little bit more difficult or probably also more subjective. But what we could see is really that regardless pro or antioxidant, somehow there was like a beneficial effect somehow in modulating the so either with hydrogen peroxide or with the two antioxidants, we observe an increase, especially in the root regeneration, which is critical in this species. So then there is another, open questions, why, what they're doing, and but I don't know if this answers your question. The concentration that we chose was based on some effects on germination, and they were significant statistically at the very early stage of germination, so something like the first 7 weeks, and then they were all plateauing, because I guess we tried to refresh the solution, but they are not so stable over time. Thank you.
Thank you. And now question from Marcin Dederski.
I have a very short question to Magda. In your plot, you showed a map of the genders of white poplar, and there's some kind of aggregation. Do you plan some spatial analysis of aggregation in the future?
Yes, I have that in plan, but yes, the work is in progress. I still take data, like the dendrological data, so yeah, I want to make some maybe model in future, yeah, because we have like very good GPS coordinates taken last week. So yeah, probably specialized it will be.
Great. I cannot wait to see.
I'm sure.
Are there any questions? If not, thank you very much to all our speakers for very interesting presentation and giving us opportunity to see first time very interesting results. And now I wish you a very nice time during barbecue party and also very emotional scientific or maybe not only discussion, and have a good time. Thank you very much.